YouTube is live? Okay, great. Welcome those listening on YouTube channel. I hope we are audible and visible there.
Yes, very good.
Okay, great. Lovely. Thank you. We'll be beginning in a few minutes. Please hang on. I still have to get a confirmation from Zoom participants. Zoom is okay? Okay. So we got a feedback from Zoom also. So both Zoom and YouTube seem to be up. If not, somebody
Yeah,
message me. Thank you.
very good.
Thank you. Thank you, Professor Mittal.
Yeah.
To all the delegates. Again, very good afternoon to one and all present here. I hope every one of you have had a wonderful lunch. On behalf of the organizing team, I, Sampriti, extend my warm welcome to all the delegates, faculty members, teachers, and young students to the session 1 of 95th NASI Annual Session and Symposium. It is our privilege to welcome Professor Sanjay Bihari, Director of SCTIMST Thiruvananthapuram, as the chair of session 1 of the 95th NASI Annual Session and Symposium. Professor Bihari is an eminent neurosurgeon with an outstanding academic and clinical career shaped through his long tenure at SGPGIMS Lucknow, his honors in medical training at SMS Medical College, Jaipur, and esteemed recognitions such as FRCS Edinburgh, the International Fellowship at AANS, and the Fellowship of Academy of Medical Science. His numerous awards and extensive portfolio of international publications reflect his pioneering contributions to neurosurgical science and patient care. Sir, we are deeply honored to have you with us today. We warmly invite you as the chair of session 1. Can I please have a big round of applause for Professor Sanjay Bihari? Thank you. Without further ado, now I would like to take the opportunity of introducing the co-convener of 95th NASI Annual Session and Symposium, Professor Jayes R. Bellare. We are honored to welcome Professor Jayes R. Bellare, former professor and an emeritus fellow in chemical engineering at IIT Bombay and the General Secretary of NASI. Professor Bellare is a distinguished scientist whose work spans microstructure engineering, ultramicroscopy, nanomaterials, biomedical devices, and advanced polymer interfaces. An alumnus of IIT Bombay and the University of Minnesota with postdoctoral research at UMass and MIT, he has made influential contributions through extensive publication, internationally recognized patents, and impactful collaborations with both academia and industry. His achievements have been honored with awards as ICI IICHE Excellence Award, NRDC Invention Award, and several major international distinctions. Widely respected for his scientific depth, Professor Belade continues to play a key leadership role in the scientific community. Sir, we are fortunate to have you among us today. Can we have a big round of applause, please? Thank you. We are fortunate to have Professor Venkata Subramaniam Ganeshan, Professor of Psychiatry and Consultant Psychiatrist at NIMHANS, Bangalore, as our another co-convener of 95th NASI Annual Session and Symposium. Professor Venkat is a leading clinician scientist in schizophrenia research, neuromodulation, and translational psychiatry. He heads the Translational Psychiatry Laboratory at the Neurobiology Research Center and leads major initiatives at NIMHANS include Instant Program for Schizophrenia and Wiser Neuromodulation Program. His work spans clinical neurobiology, metabolic abnormalities in psychiatric disorders, TDCS application, and advanced brain imaging. So we warmly welcome you with us today. Can we please have a big round of applause? Professor Belade, now I would request you to come up and...
Thank you. Thank you very much for that introduction. I'll just take a minute to put this in context, So, Professor Venkat will give us more details on the genesis of the particular topic, but let me just share a little bit of background of how we do these things, right? Often this symposium, we do it on a yearly basis, NASI does it on a yearly basis. There's a lot of discussion in choosing a topic, and we try to, in general, we try to broad base this topic so that every specialty is covered, right? So for example, in the past we've had programs that said, how does the scientific community contribute to Viksit Bharat? And we had sessions across all possible disciplines. And in general, that's how it has been. But this year is different. And it's different because the theme of this is virtually mono-focused, right? It's focused on one important aspect of, I would say, life, which actually troubles us all. As a professor, I see many troubled students because of stress, family situation, interpersonal relationships, all sorts of reasons. Come up with, have these mental issues. There's another set of technologies which, as an engineer, I'm very excited about, and that is the brain-machine interface. Invasive and now non-invasive, right? Remotely, somebody's reading my mind thanks to a probe that is reading, MRI or other signals from me. Right now, it has to be in proximity, but I'm sure it will be done remotely soon enough. And the third aspect is where the brain and its circuitry is being revealed, on the one hand through microscopy and other techniques, and also where this is now inspiring technology. Of course, it's inspired AI, and there the neuro angle is well known, but it's also inspiring now circuits, microelectronic circuits, where neuromorphic kind of computing is being done both on the software side as well as on the hardware side, where circuits are inspired, and circuits dynamically change on a chip. So this is some of the influence of brain research and mental health and cognition, and these are complex technologies which I think require discussion. And Professor Mishra has led this effort in putting together this program along with Dr. Venkat. And I leave it to you, sir, to please give us elaboration of this before we begin with the talks. Thank you.
Thank you, Professor Belade. Now the mic is passed to Professor Venkata Subramaniam ji. So please.
Thank you. Good afternoon all. And at the outset, I really feel humbled and privileged to be here, especially for this symposium. The thematic focus was quite aptly chosen by Professor Misra. When Sir approached me with this topic focus, it was fascinating the way in which he had envisioned how the program should be constructed. In that context, we had some discussions with Professor Bellare, where we had thought of putting the focus to involve the hardware, that is the networks. So that's how it came up in the discussion. And then the software, which is the cognition and its aberration in terms of disorders. And then how different of us from various specialities can come together, whether it is neuroscientists or engineers, clinicians, entrepreneurs. So how we can make the brain health and the disorder better in terms of understanding it more. I think Professor Mishra briefly touched upon us in the morning talk. I think about a decade or two before in the journal Science, there was a set of questions. What are the top 20 questions that should be addressed? The first is the nature of the universe. The second compelling question is, what is the nature of consciousness? So that's what you alluded to, sir. So the brain is so complex, and this symposium theme aptly reflects that. And to add a few more points of what Professor Bellare mentioned, the sessions span across all these aspects, right from the cognitive networks and how it can be modulated, and how we can collaborate with entrepreneurs from various backgrounds to translate it. So that's how the entire symposium flows. To end, I just want to make the quote, the famous quote of Lyle Watson, which surmises the complexity of the brain. The quote goes like this: if the brain were so simple we could understand it, we would be so simple we couldn't. So with these few words, I again thank Professor Mishra for this opportunity and Professor Bellare. I think together they guided a lot, and I was just an assistant which kind of put certain things together. Thank you.
First of all, it's an honor and privilege for me to be here as a part of NASI, and I'm grateful to the executive committee for actually giving me this honor even before I'm a part of the actual thing officially. So, it's a beautiful title, Healthy Brain, Mind and Cognition from Computers to Clinics. And let me first focus on the first talk. So from an end user experience, let me take you back to the research. So when we are actually making any movements of the body, the first thing is we have the pyramidal system, which is actually guiding finer movements of our body, right? Finer movements is being guided by the pyramidal system. Then there's an extra pyramidal system, which actually supplements all the other functions which guides the finer movements of the body. This is the extra pyramidal system. And then there is a predictive system. This predictive system is run by 4 different areas. The first of them, of course, is vision. So vision provides you the balance, it provides you the pathways, it provides you the dangers which you are going to encounter on a daily basis. The second important thing is the vestibular system. So when we rotate our head, when we flex our head, when we lie down, when we stand up, how do we maintain our balance is actually guided by the vestibular system. The third very important system is the cerebellar system. And what cerebellum does is to predict what the next step is going to be and smoothen out the movement in such a way that they appear to be absolutely smooth and full of coordination. And finally is the vestibulospinal tract, which actually coordinates all our muscles to do a particular movement. And I think there's no person better than Dr. Vatsala Tirumalai, who's the Professor and Dean of Research at the National Center for Biological Sciences, Tata Institute of Fundamental Research at Bangalore, to talk about this wonderful work on cerebellum and also using predictions in the brain for predicting behaviors. That's a very important topic that she's going to talk about. And she earned her B.Tech in biotechnology from Anna University and a PhD in neuroscience from Brandeis University. Her cutting-edge research on neural circuits controls movements in zebrafish. This has produced 30 plus high-impact publications. She's a recipient of Shanti Swarup Bhatnagar Prize of 2020. She's a senior fellow of the Wellcome Trust TBT India Alliance. She also leads BioWise, championing women's leadership in science programs. So we are truly honored to have her with us, and I think she will give us a lot of information on the role of cerebellum in actually predicting our motor movements. Thank you.
It's covered there. It's also shared on. It's shared on Zoom. Okay, good. Okay, good afternoon everyone, and thank you to the organizers as well as the chair of the session for such glowing words about me. I hope my talk lives up to those words. I'll try my best. So what I'm going to talk about today is how the brain generates first of all, and then uses predictions to guide behavior. And as was referred to, I will be using, or I will be talking about the fish model system. See, as biologists, we take inspiration from everything around us, be it a very simple yeast or humans. We look at the complexity of each organism. Even if you take a single-celled organism like a yeast, there is a lot of complexity in that organism, and it can be used and has been used for understanding various biological processes. So in that sense, a fish, as you might see around you, might simply be swimming around, but its brain is capable of a lot of complex computations which helps it survive, find food, avoid predators, and so on. So the work that I'm going to be talking about is primarily the PhD research of two very brilliant graduate students, Shivangi Verma and Sriram Narayanan, with inputs from other lab members as well. So making predictions is an inherent, oh, it's not moving, it's not going forward there. It's also not going forward there, if I, yeah. So, whether you're standing at a traffic light waiting for the light to turn green for you to go, or you're playing performance sports like tennis or golf, predictions from the brain guide your behavior. And we do not think about these things because these are happening at a subconscious level. Okay. So, even in the natural world, animals, when they use predictions, they are calculating, for example, for the important function of finding food, the behavior of the fish, how it is going to jump, is being predicted by the bear, and only if it puts its mouth right in the future position of the fish, it will be able to catch it. And these are examples of how the brain uses predictions to guide behavior. And neuroscientists for long have studied these behaviors and studied predictive processing in the brain in the context of many different tasks, including sensory encoding, when you're seeing repeated sensory stimuli in your environment, in terms of reward, because reward is important for all of us. And we predict when the reward is going to come, how much reward is going to come, and we calibrate our actions based on the reward. The more the reward, the more higher risky behavior that we see around us. For example, in gambling and in lottery tickets, in horse racing, and so on. So predictive processing is all around us, but unfortunately we understand very, very little about how predictive processing works. What are the brain regions involved? We know a little bit, but how exactly these brain regions are guiding the predictive behaviors
The
is something.
PowerPoint sharing would be better, sir. I mean, let's go.
Sorry?
I mean,
Okay, let's see. Can you see this?
yes, sir.
Okay. So, the cerebellum is a very highly conserved structure of the brain. It is pretty much found in all vertebrates, and it has stayed the same over 450 million years of evolution. If you look at the cerebellum of a fish, and if you look at the cerebellum of mammals, fish and mammals diverged from a common ancestor about 450 million years ago, but still you will find pretty much the same circuitry in terms of the principal neurons, which are the Purkinje neurons, which receive inputs from two major excitatory pathways, the parallel fibers from granule cells and the climbing fibers from an extracerebellar structure called the inferior olive. More about this later, but because the cerebellum is such a conserved structure, we can study predictive processing and the cerebellum function in early fish such as the larval zebrafish. The larval zebrafish are suited for this exercise because they are independent organisms as early as 5 days post-fertilization. So here is a larva that is only 5 days after the egg and the sperm fused to form a zygote, and it's already swimming around in its environment. It responds to visual stimuli, it avoids predators, and it also hunts for its own food. Contrast this with humans who probably take 20 years or longer to become independent. The other major advantage of using the larval zebrafish is that its brain is transparent, so you can look at the neurons and record their activity through a microscope without having to perform any invasive surgery. And that's what we use in this work. So, here is an optomotor behavior, which is an innate reflex response, and fish use this to maintain their position in a flowing stream of water. So these fish live in the Ganges and its tributaries as well as in Brahmaputra. Actually, a lot of them can be found in Assam and in northern West Bengal, and because they are so small, they can be washed away by the current. So they constantly look at the world around them, and if they see that the optic flow is receding from them, imagine yourself standing on a treadmill, and if you're just stationary, while the treadmill moves you backwards, the scene around you will move forward. And to compensate for that, if you keep moving forward, then the visual scene remains stationary, and that's what the fish is trying to do. So here we put a larva under a spinning disc, and the disc is showing black and white patterns, and it's moving along. Oh, it's not moving forward. It's not moving forward over there. Shade window is closed. What happened? Is it shade on the meeting?
I don't think there's no need to share because.
Is it okay if it's not shared on Zoom? No, it's not. Okay, I'll keep talking just in the interest of time. So, optomotor response helps larvae stabilize their position in a moving water body. We can recreate it in the lab by showing them black and white patterns underneath them, so they can see this and then they can move according to the in the same direction of the black and white stripes. We can also recreate it in the lab in head restrained fish, because remember I said we'll do microscopy to look at the brain. So we want the fish to be stationary, at least the head to be stationary in one place. So we embed the larvae in agarose, and then we use microscopes to look at the activity of the neurons. अभी तो वर्क कर रहा था ना, क्या हुआ? Okay. So, is it going on Zoom? Okay. Okay, good. Okay, so that is the behavior of the larva, and here is the head restrained larva. Also, when you show the black and white grating, it is so the question is, can a simple cerebellum do predictions? So we used optic flow stimuli, and we created multiple instances of optic flow to practice them, and we call these acclimatization. And what happens is that we present them multiple forward-going stimuli, optic flow stimuli, and then suddenly surprise them with something that they're not expecting, which is a weak backward-going stimulus, as would appear right now. So the gratings are actually shifting backwards instead of forward, and this is something that the fish is not expecting. And the question is, do fish learn from the acclimatization stimuli? When you give multiple instances of the forward-going
Recording
stimulus,
in progress.
does the fish learn that this is the pattern of the sensory stimulus that it's going to receive, and then when it receives the unexpected surprise stimulus, does the brain show any signatures of error, that this is not what I was supposed to be receiving? So that is what is represented over here. So here I'm giving 8 instances of forward-going stimuli and 3 instances of the probe stimuli, as is shown here. So what we find is that as you give multiple instances of the forward-going stimuli, the latency to generate a swim response goes down. That means that the fish is able to respond faster because it already predicts, it already knows what's going to come next. But when you give a probe stimulus, the weak backward-going stimulus, it gets surprised. First of all, there is no motor response for the probe stimulus, but the next forward-going stimulus after the probe stimulus, the fish is sluggish to respond. So the latency to generate a swim response goes up after the probe stimulus, as is shown here. And if you lesion the cerebellum, this effect goes away. That means the cerebellum is needed for updating the predictions about the world.
Recording
and
stopped.
when the cerebellum is no longer there, then the updating of the stimulus status from the outside world is upset. So we wanted to look at this a little bit more in detail, and the way we did that was to use 2-photon imaging of transgenic zebrafish, which express a calcium-sensitive protein in Purkinje neurons of the cerebellum. So these neurons shown here in the panel below are Purkinje neurons expressing GCAMP6S, which is a genetically encoded calcium sensor. And when the neurons become active, then the neurons glow. Their fluorescence goes up, and you can measure this, and you can quantify this, and you can tell which neurons were activated by what stimulus and when. So that this is the movie one more time for you to see that. And when we are doing that, of course, we are showing them the repeated optic flow stimulus, just like what I showed before, the acclimatization stimuli of multiple steps, 8 8 repetitions, and then the probe stimulus followed following the acclimatization stimuli. And here, that is the setup. And before we can look at the Purkinje neurons, it's also important to appreciate that not all Purkinje neurons are the same. There are Purkinje neurons tuned to different kinds of parameters. There are Purkinje neurons which respond only to motor bouts when the fish is swimming. There are Purkinje neurons which are sensory tuned, and they are tuned to sensory stimuli of different directions, so forward direction, backward direction, and so on. So it's important to determine their tuning properties. And for that, we present randomized stimuli of forward and backward of varying strengths. And then we ask, using a regression methods, we ask whether the Purkinje neuron is motor tuned. I won't go into much detail here in terms of, in the interest of time. So are they motor tuned? Are they activated by forward going optic flow stimulus? Are they activated by backward going stimulus? These are the probe stimulus that I talked about, the stimulus that is causing the surprise. And we'll focus in specific on the backward tuned Purkinje neurons, because these are the neurons that encode the surprise value. So what we found was that when you give 8 repetitions of forward going flow stimuli followed by the weak backward going stimulus, the Purkinje neurons show a surprise signal that's very, very large. So much enhanced compared to baseline. Baseline is before they were trained on these forward going stimuli. So there is this we call as the probe response or the predictive error response, because there is a prediction that's being generated, and when the prediction is not met, then these neurons generate an error response. Now let's look at these properties of this error response a little bit more. What we find is that the error response is only enhanced when the forward going stimuli are given multiple times. 2 times does not do it, 4 times does not do it, but 8 and 16 times, then the responses are enhanced to a very significant level. And there are many other things that we report in this study that was published last year, so I'm not going to go in great detail about these predictive error responses and all of their different signatures that we reported in this paper, but I'm going to dwell on the importance for behavior. So I talked about swim latency, and what we wanted to know was whether these predictive error responses that we record in the fish Purkinje neurons is in any way correlated to behavior. And because we're doing calcium imaging, we can look at multiple neurons at the same time. And at a time, we are able to look at up to 60, 70 neurons in a single plane. And when we did that and we looked at the distribution of predictive error responses, we find that in those trials where a bunch of Purkinje neurons are showing very strong predictive error responses, those fish are capable of producing movements quickly. And in neurons where the predictive error response is weak, and in fish where there are not very many Purkinje neurons that are encoding the error response, then in those cases the fish are slow to respond. Okay, so this is a direct connection between, I showed you the lesion data before, where if you lesion the cerebellum, you get absence of the latency modulation. Now, looking at the activity of Purkinje neurons, if the Purkinje neurons are very active during a probe trial, then the fish does well in swimming. But if the Purkinje neuron is not so activated by a probe stimulus, then the latency is large. Sluggish response. So strong expectation, short latency to swim, and weak expectation, long latency to swim. And this can be quantified here, where there is a strong negative correlation between latency and the prevalence of the predictive error response. So what we wanted to next ask is, okay, these are calcium responses. In general, there is a lot of calcium that comes into neurons whenever they fire an action potential, when they become activated. But Purkinje neurons are also special because they receive a synaptic input that also brings in a lot of calcium. And this is the synaptic input that comes from the inferior olive into the Purkinje neurons, and these are carried by what is known as the climbing fiber. So here are simultaneous recordings from Purkinje neurons while we are doing the calcium imaging, and we show that these tall ones are the climbing fiber inputs, the tall lines, and those lines correspond to very strong calcium signatures in these neurons. So to delineate what is exactly happening in the Purkinje neuron in terms of its electrical activity, Alok Verma, another graduate student in the lab, performed recordings from single neurons while the fish was executing these behaviors. So we showed them the visual stimulus, we made them swim, and then we looked at the motor response as well as the activity of single Purkinje neurons by means of patch clamp electrophysiology, and we find something very interesting. What we find is that the activity of the Purkinje neuron begins to ramp up even before the trial has started. So this is predictive, right? So it ramps up, and then once the visual stimulus starts, it goes boom, the activity increases a lot. And that is what is shown here. These are the firing rates, and you see that both the spiking, which is the smaller unit, as well as the climbing fiber synaptic input, both ramp up before the start of the trial, and then it increases massively after the trial starts. And not only that, if you look at the strength of this ramp and correlate it with the latency, again you see a negative correlation, which means that when the strength of the predictive ramping signal is high, then the latency is small, and vice versa. So there is real predictive value in the Purkinje neuron activity even before the visual stimulus starts. And in the last few minutes, I will tell you about a recent work that is also under the publication pipeline, where we're looking at this climbing fiber input. And I want to mention this work because it also has relevance for the topic that we are talking today in terms of healthy brain. And we know that there are many conditions where neurodevelopmentally there are deficits that happen. And one of the deficits that happens in autism, for example, is the deficits in synaptic pruning. Synaptic pruning is the loss of synapses. We all know about synaptogenesis, which is the formation of synapses, but synaptic pruning is equally important. And in the human brain, actually the adult human loses up to 50% of all synapses formed. And if this process goes awry, if the synapses are not pruned at the right time, then it can lead to circuit deficits, and it can lead to conditions such as autism spectrum disorders. So, but the thing is that we still don't understand what happens to the circuit when synapses are pruned away. And the zebrafish system is again a very nice system in which we can probe what happens when synaptic pruning happens, what happens to the circuit. And so that's what we undertook, and we used the same predictive processing paradigm, and we asked, in the Purkinje neuron, we know that early on there are multiple climbing fibers innervating a single Purkinje neuron. Over the course of development, this polyinnervation becomes monoinnervation, such that in the adult animal, there's only one climbing fiber that innervates each Purkinje neuron. So there's a one-to-one relationship. And we asked, how does the processing in the cerebellum change when the cerebellum goes from polyinnervation to monoinnervation? And to cut a long story short, what we saw using the same paradigm, the predictive processing paradigm, is that the predictive error responses, which are enhanced in late larval stages, when there is only one climbing fiber instructing each Purkinje neuron, is much better compared to the early larval stages, where there are multiple climbing fibers innervating each Purkinje neuron, and it causes confusion for the larvae because it doesn't know what this synaptic input means. And I'm not going into the tuning of these different synaptic inputs in the interest of time, but we find that, so the green is early stage larvae with polyinnervation, and the purple is the late stage larva with monoinnervation, and then there is an intermediate stage with gray. Over the course of development, the probe response is enhanced much more in the late stage compared to the early stage, and the behavior is also modified accordingly. So the early stage larva, where each Purkinje neuron receives multiple climbing fiber inputs, is unable to modulate its latency. That's what that graph shows over there, where the lines are not showing very well. I don't know, the light colored lines are not showing very well. Whereas the late stage larva with with mono-innervated Purkinje neurons is able to modulate latency. So it's able to learn its sensory environment. It's able to understand that this is the pattern of sensory stimulus and modify its behavior according to what it sees on a moment-to-moment basis. So I will stop there, and this is the summary of the work. It's available as a preprint, where we show that the sensory tuning as well as the motor error tuning of Purkinje neurons changes over the course of development, and this affects how predictive error responses mold behaviors. And with that, I will summarize. I've shown you that fish can learn to expect optic flow stimulus by estimating probability of occurrence based on prior experience, and olivocerebellar circuitry implements an error feedback, and the cerebellum is involved in this process, and the work identifies a new role for the cerebellum in predictive processing of sensory inputs. Finally, I also showed you how synaptic pruning plays an important role in this process. And I would finish by thanking the funding agency and also all my lab members, the students who contributed to this work, and collaborators for providing reagents. Thank you.
Thank you, Dr. Vatsala, for this wonderful presentation. I just wanted to first of all, highlight the clinical significance of the work that you have done. So, cerebellum has several types of cells. One are the granular cells. The granular cells are the sensory input cells. So, they get all the sensory information from the entire body. Then you have the Purkinje cells, which actually correlate with the cerebral cortex. And all the input which comes out as coordination comes to these cells. And then you have stellate, basket, and Golgi cells, which actually are interneurons and other cells. So this is all. So what is the significance of this study? At some point in the future, you could there are several diseases, like you have spinomuscular atrophy or cerebellospinal atrophy, where the Purkinje cells are gone, so you could kind of modulate that. Then you will have tumors of the cerebellum. When you remove those tumors, many of these problems come in. And the third is infarcts, posterior circulation infarcts, where part of the cerebellum actually dies. And the fourth is, of course, you have Chiari malformation, in which the cerebellum becomes atrophic and comes down into the spinal cord. So in all these areas, these work will be very important. And because the future is that cerebellum controls three things: balance, muscle tone, and coordination. So if you are getting one Purkinje cell and one sensory input into that Purkinje cell, maybe at some point in the future, you can actually control how the patient takes care of coordination, balance, and muscle tone. So thank you very much. That's a very wonderful thing.
Thank you.
Yeah, very fascinating presentation, Professor Astra. I have two questions. One is, now more and more prediction seems to be, some kind of common connect, and brain is conceptualized as a prediction machine.
Yeah.
So the first question is it unique to cerebellum or cerebellar neurons? So that is one. The second related question is, I was quite fascinated about the last study that you described, because especially alluding to neurodevelopmental disorders like autism. So autism is one condition where certain parts of the brain is larger. Generally we find brain deficits, especially cerebellum is reported to be larger in children and adolescents with autism, of course. So do you have any comments on that? Is it? Yes.
Yeah, so to the first question about whether it is unique to cerebellum, no, it is not. Predictive processing is actually widespread. That's why I said, brain is a predictive machine, because it does predictions in multiple places. In the cortex, in the basal ganglia, for example, reward prediction is very well known, that the basal ganglia is very important for predicting rewards when you're undertaking a task. And the cortex, of course, is important in predictive processing of multiple things, sensory as well as motor. When it comes to cerebellum, it was not appreciated as a very major predictive machine because it was thought to act at the millisecond time scale. It was not thought to act at the behavioral or the seconds and minutes time scale. This study shows it. That's another thing, that we show it at the seconds and minutes time scale. And the reason for that is that the cerebellum initially was studied in the context of eye blink conditioning. You know, you puff an air in front of the animal and you play a tone first and then you puff the air. It will learn to blink its eye even before the puff of air comes. But that is on the 500 millisecond, up to 500 millisecond time scale, not more than that. These different regions are there. They all do predictions, but they do predictions under different contexts. And there are also times when all of them work together. For example, recent studies also look at cerebellum in terms of reward, that there is reward prediction happening in the cerebellum as well. So there are all these loops, cerebellothalamic, cortical, as well as cerebellothalamic basal ganglia loops that, keep processing the information, and it's not clear exactly who does what. In terms of the second question for the neurodevelopmental aspect, the autism spectrum disorder, at least basic neuroscientists understand autism spectrum disorders as arising out of deficits in EI, excitation inhibition balance. If you look at the literature, different people will tell you EI balance is upset in favor of excitation or in favor of inhibition. My take is that it's just upsetting from the balance. It doesn't matter which way it goes. It may go more excitation or more inhibition, and that could happen precisely because the synapses are not pruned properly, and you have more excitatory synapses or more inhibitory synapses.
Yeah, I'm not a neuroscientist, so my question will be totally different. You said removing cerebellum removes absence of response. The surprise response is removed by removal. That's a conclusion drawn by one removal. Do we know, are there any global deficiencies in addition to what you looked at? You looked at one aspect.
Correct.
What about other aspects?
I didn't put that slide here. So that's a very good point. So when we use lasers to ablate the cerebellum, and then we look at the basic locomotor function of the fish. Is it able to swim properly? Is it generating the appropriate number of swim bouts in a minute? Does the swim bout look normal, as in terms of how it swims around in its environment? And that looks normal. That
So
is
phenotypically,
it is normal. Yeah.
And another point is that your polyinnervation goes to monoinnervation, and that is somehow related to autism also, you mentioned. But isn't it autism a childhood incident, but this process happens at adulthood? But the autism reflects in the childhood. So how the time scales are matching?
So I'm not making an autism model here. It was a motivation for us to look at the functional outcomes of synaptic pruning. Why do circuits make more synapses than is needed and then they prune it away, right? And when they prune it away, what happens to the circuit, right? Because if you look at a human infant, for example, as the infant grows into toddler and then a young child and then an adolescent, there are two major waves of synaptic pruning, one happening in the toddler years and then one happening in the adolescent years. And both are important. And this will upset the brain because suddenly massive numbers of synapses that were previously exciting your neuron are not there. How does the brain change its function when this happens? That has not been explored, and here we show a behavioral output of that. That is the point here. Yeah.
So thank you for this wonderful presentation and all the best for the future. I think you have a lot of future possibilities in this research. Thank you.
Thank you.
Thank you. A big hand of applause for that. I'd like to just share with you the second presentation, which is equally important. And here we are looking at mental health. So according to the World Health Organization, almost 1 in 7 people have some kind of mental health. So a health issue. So I think there are several people in the room itself, they may not recognize that they are suffering from mental health. It's that important. And the other important aspect of mental health is the caregiver response to mental health. because so little is known about mental health that it's really traumatic for the caregiver when you are confronted with a patient at home whom you don't understand anything about, and yet you have to manage that patient, and there is this huge social stigma attached to this patient. Now, when we look, what are the problems associated with mental health? One is that you have major psychosis. So you have very clearly differentiated things like schizophrenia, bipolar disorders, severe depression, post-traumatic stress disorders, or eating disorders like bulimia, or you have neurodevelopmental disorders. They are clearly differentiated, and you can actually make out a patient who has these disorders. Then there is a second one, which are neuroses, which are very subtle early disorders, where there may be depression, anxiety, there may be a little bit of obsessive compulsive disorder, there may be a little bit of anxiety. And of course, when you look at the entire spectrum of population, then some of them may be having some autistic behavior, which you may not actually come to know about. And there may be problems related to cognition, there may be problems related to emotion, and there may be problems related to behavior. So you can understand, and this could be primary psychiatric disorders, and this could be secondary psychiatric disorders. So you can imagine the complexity of work which is required when you're actually looking at these disorders. And I'm very honored to have Dr. Arjun Ramakrishnan, who's coming online. He's the assistant professor of the Department of Biological Sciences and Bioengineering, IIT Kanpur. I personally visited that lab, and they're doing really fantastic work on bioengineering and biological sciences. So he holds a Bachelor of Engineering degree in telecommunications from BMS College of Engineering and a PhD in neuroscience from National Brain Research Center, Gurugram, followed by postdoctoral research at Duke University and the University of Pennsylvania. He's a recipient of NARSAD Young Investigator Award and the DBT Wellcome Trust India Alliance Intermediate Fellowship. He has published extensively in systems neuroscience, decision making, and neurotechnology. Through his innovations in neurotech workshop and outreach initiatives, he continues to promote accessible neurotechnology and foster scientific awareness across India. And his topic is very interesting. Rather than focusing on these different types of psychiatric disorders, he's talking about neuroengineering in mental health. Thank you.
Good afternoon, everyone. Thank you, Professor Bihari. I mean, this was a fascinating introduction to what I'm going to say. Again, I really hope all of this lives up to the introduction as well. With that, I'll quickly begin the talk. And it looks like we are running slightly late, so I will get started right away. I hope the slides are visible to everybody.
Yeah, visible.
Okay. Excellent. So visible in the audience as well?
Visible online.
Visible online. I'm not able to hear from the auditorium actually.
Sir, you're audible.
I'm audible? Okay. Okay, now I'm able to hear. Thank you very much for confirming. It would have been great to be there, but unfortunately I'm sitting several thousand kilometers away in Kanpur. And even though I'm several kilometers away, being there virtually for a talk can also be somewhat nerve-wracking. And I'm actually having like cold hands and cold feet, and my heart is racing. And I'm constantly thinking about how I can proceed with this or what might go wrong and so on. And this is sort of the worrying feelings that people sometimes or many times go through. And in some people, in some occasions, becomes really hard to suppress. And those are core symptoms of today what we call as anxiety disorders. So I'm going to take you through what anxiety disorders are, the gaps in our healthcare, the new ways that we are approaching this condition, and a series of experiments to convince you that we are able to capture and quantify some of the things that we are interested in. So what are anxiety disorders? So DSM-5, this is the Diagnostic Statistic Manual. Version 5 is the latest version of this. Characterizes generalized anxiety disorders as excessive anxiety and worry for more than a period of 6 months. And these are worries that are sometimes very hard to control, like I was talking about in the beginning, along with certain other symptoms like restlessness, being fatigued, difficulty in concentrating, irritability, even including sleep disturbances. A large number of people, about about 500 million people worldwide, suffering from, according to a latest survey from Lancet, are suffering from anxiety and depression. This is around COVID period when these numbers actually spiked up a lot. And if you look by age, these numbers are dominated by people in the 25 to 40 age group, and a majority of them are actually women. However, what are the gaps in this? Even today, we are using a trial and error approach in treating people with anxiety. We lack what is a very purely objective measure of this condition. And there is a lot of messiness because of overlap with other conditions and other symptoms and comorbidities. And the neural basis of these disorders is still not entirely clear. So what we would need, ideally, is something like a thermometer that tells us that someone is suffering from a fever of a certain degree, and tells us what what they're really undergoing, or be able to quantify the severity of the symptoms, so that we can reduce the time to recovery, so that we can have better diagnostics to catch them early, and so on. And what we are trying to understand is use decision making to really understand what is going on in these conditions. So a talk like this, for example, is an environmental factor. So it puts you in a state of anxiety. Whereas if you have a biological predisposition, along with a continual onslaught of stress in your early life as well as later into life, this could predispose you to what is called or operationalized as trait anxiety, which is a continual state of worry. And these people who are trait anxious have a very high chance of converting into those diagnosed for anxiety disorders later in life. So the question really is, if you're trait anxious, can we really pick that up by looking at how you make decisions? And this seems intuitive, because if you're anxious, if you're worrying about things, it's possible that you veer off the script and say things that you don't want even in a talk like this. A very standard probe that has been used across many labs and over several years is what is called a dot probe test. This is actually very simple. All that people have to do is report whether these dots are horizontally aligned or if they're vertically aligned. However, before they see the dots, they flash this sort of an emotional face sometimes, or a neutral face, and this seems to hijack their attention in such a way that even if they report correctly, they end up taking a lot of time. So this was a very standard assay that was used to understand anxiety. Although this taps on some natural aspects of anxiety, high emotional reactivity, it seems to have had some inconsistencies, and things have not been consistent across different labs and applications, which has warranted further investigations, better understanding of the underlying mechanisms, and a more naturalistic task that can capture what is really going on in anxiety. So what we are going to do is tap on what is called hypervigilance. So anxiety taps on hypervigilance, which is your tendency to remain alert all the time. So for example, imagine being in a forest camp where tigers are at large, and you have to do something on your phone, and you're going to be constantly paying attention to sounds that are coming from all around. And to do that, you have to disengage from your phone. And this promotes a lot of quick learning and updating based on the sounds that you're hearing. If you hear an actual tiger, you might as well pack up and run. So this is really what we're tapping on. To do this, we are using a neuroethological approach, and we have designed a task that is based on what is called patch foraging. So why patch foraging? I mean, this is a very naturalistic task that is conserved across species, humans, animals, including monkeys, rats, and you can go further down, like all of them forage for food. So this is something that is very, very natural. And in fact, there is a strong hypothesis going around that the primate brain size has increased based on foraging complexity, going from lemurs to baboons to humans. And also, this sort of a paradigm provides you a normative framework. So it tells you that as long as there are rewards, you might want to stay in a certain patch while getting some berries. Whereas as soon as this is not rewarding enough, you might want to move away. But this balance between reward and travel is actually, there's always a sweet spot that you might want to find, and something that people and animals naturally do to be able to survive in these conditions. And we and others have actually also recorded from different brain regions, including what is called the anterior cingulate cortex. So this is a sagittal section of the human brain, and this is the cingulate region, and this is the anterior part called the anterior cingulate cortex. And what we have found is that neurons in these regions are actually signaling how long you should stay in a patch. But what is really interesting for the current talk is that these neurons in the anterior cingulate cortex is also tightly connected with another region in the brainstem called the locus ceruleus, and these send norepinephrine signals up to a large part of the cortex, including the ACC, and this circuit, especially the locus ceruleus norepinephrine to ACC circuit, is highly implicated in anxiety and as well as in foraging. So, we thought that there might be some kind of an overlap that is very intuitive for us to pick on. And coming to a very conceptual understanding of what might be going on and why patch foraging is an interesting approach, it might capture an anxious mind, because disengaging from a patch is perhaps equivalent to disengaging from a thought, or disengaging from your smartphone when you hear a growl in the background. So to understand really whether this is all going to work, we set up a virtual foraging task in the lab. So essentially people had to collect fruits, and at some point they decide that they want to abandon this to go to the next patch that is over somewhere in the background. So the task actually proceeds in a way like this. It's a very simple task where you go to a patch that you're nearby and you collect some berries from there. But as you go there often, the amount of rewards you get reduces, because you've picked all the good ones and you're left with very few berries, or the bad quality ones. And so the amount keeps going down, sort of mimicking the natural condition. And at some point you decide that it's not worth going back to the same patch, and you indicate by clicking on the square that you want to go to the next patch. So this is really what is happening in this game. And what we see, and we've done this in monkeys, we've done this in humans, and we have a lot of data. And what we see is very interesting. With very less training, you see that humans and animals are near optimal in these conditions. So they know exactly how long to stay in a patch and when to leave. So for example, this is data from monkeys that played this game, where the distance between these patches was different in different conditions. So if the next patch or the next tree is very far away, then you might as well stay in the current tree for longer. And that is exactly what is signaled by this increasing trend here. And you can see that one of these lines, this green line here, is actually the optimal leave time computed based on the distance that we've calibrated. And monkeys actually very closely follow that optimal trend. And in fact, this is true for humans as well. So this is data from a large number of individuals who played this game. And you can see on average here that the optimal stay duration is about 50 seconds in a particular patch. And you can see on average, they are hovering around the 50-second mark, even though you see that there is a lot of individual differences, which is actually what I will come to next. So one thing that you might have noticed in this sort of a setup is that you have to repeatedly click on the patch and click on the square to actually proceed with the game. So one might naturally think, and this is sort of akin to picking the fruits and so on, that a large component of this act includes how quickly you respond. And so there might be individual differences in response times, and maybe that is what is factoring into the individual differences that you see in behavior. And it may or may not have anything to do with anxiety. So we actually quantified the inter-individual differences in response times, and there was actually quite a bit of difference. So you could go from 2.5 seconds, someone who's playing very fast, to something about to somewhere like 4 seconds between trials. And this actually does change how rewarding the particular environment is. If you're very fast, it is actually more rewarding. But then you do see that people adjust their behavior according to their responses. So if you are a fast person, you also stay for lesser time. And if you're slow, you stay for longer, right? So all in all, these response times do matter. So models that take into account response time, so they do effectively predict behaviors much better. So what we did was we put in response times into our models, and we also factored that into the individual differences. But what we did see is that their anxiety level, so here I'm calling it trait anxiety because these are all healthy participants playing the game, is in fact still a very strong predictor of how they play the game. So in fact, as I sort of set up the framework, if you're anxious, you're thinking about other things, you're finding it very hard to engage with the task, and you disengage sooner. And that is really what we see, that if you're high anxious person, you disengage from the path sooner, because, and therefore it reflects as a much lower stay duration in general for these participants, taking into account all the other things that vary in the task. Now, because reaction times are an essential component of this, we came up with a process model for what happens in every decision that goes into this sort of a game. For that, we made use of what is called a drift diffusion model. It's actually something that goes like this. So essentially, whenever you're faced with options like option 1 and option 2, and you have some evidence for one option over the other, you take that evidence, you deliberate over it for some time, and then you make a decision that you want option 1. If, let's say, there is a lot of evidence for option 1, then you make that decision much faster. If there is less evidence, like option 1 being almost the same as option 2, you might take much longer to make that decision. So that is really what this model is conveying. And sometimes when you have to make the decision very quickly, you might actually not wait for a lot of time. You might not deliberate long enough, and you might make these decisions much quickly, depending on the urgency of the situation. So the bounds collapse to allow you to make that decision faster with lesser evidence. This axis is actually evidence, and this is time in this. Okay, so we actually use this model, flip it around to understand what is happening in the patch foraging situation. So where we called option 1 as staying in the patch, option 2 as leaving from the patch. So if you have a lot of evidence to stay in the patch is very rich, then you do that decision really quickly. If you have less evidence, if let's say leaving the patch, or staying in the patch is almost as good as going to the next patch, then you should take much longer to make that decision, right? And this is the model that we fit to the data, and it's actually a very good fit to the data and explains what is happening in the task. But what it also tells us is that those with anxiety, all else controlled for, have a much reduced slope of this decision in this task. And you look at the 95% confidence intervals, it shows you that they're clearly less motivated or less engaged in the patch that they're currently in, which explains why they're leaving the patches sooner. So these people who are high anxious, they end up leaving the patches when they're still rewarding. So that's why the instantaneous reward is still pretty high. But overall, the cumulative reward ends up being on the lower side because they're leaving too soon to actually capitalize on the richness of the environment. So this is great. So we've set up a new task. We have a computational framework to understand what is going on in this task, and it seems to be pointing to how they disengage from this task sooner. So we have a handle over how trait anxiety seems to be affecting foraging decisions. But as I said earlier, we also understand that there are some brain regions that are implicated in this, and the locus coeruleus is projecting to ACC, and this circuit seems to be important to this. But to really ascertain whether that is the case, we wanted to look at some markers of this particular circuit that we are interested in. One of them is actually pupil size, because this is when brightness is controlled, it reveals a lot of underlying cognitive processes, among which one of them is in fact the release of locus coeruleus norepinephrine cortical circuit, which also is projected to a branch of neurons that control the size of the pupil. So likewise, even in non-invasive signals that are obtained from electroencephalography, there are certain signals that are a measure of this particular circuit. So we're going to focus on one, pupil size, both baseline pupil size as well as phasic changes. There are very quick changes in pupil size. They indicate different aspects of this locus coeruleus activity. Likewise, on the EEG side, we're going to focus on what is called the theta-beta ratio, where theta seems to signal conflict. So if you're conflicted about a particular option, then there is an increase in the theta power, increase in pupil dilation as well. Whereas beta seems to signal some kind of top-down inhibition. So the ratio actually ends up capturing the balance between the reward and the regulatory control. So essentially what we hypothesized going forward was that we know trait anxiety affects decisions. We are hypothesizing that this circuit is underlying what is happening here, the dysregulation in the circuit. And we want to see whether that reflects in this sort of a signal that is known to be a biomarker for this particular circuit. If that holds, then we can implicate this circuit in this dysfunction. So essentially what we noticed was that trait anxiety does impact theta beta ratio. So you can clearly see here, focus on, I mean, these are posterior distributions, and there is P300 as well, but don't look at this one. But look at theta beta ratio here. You can clearly see that there is a very high increase in theta beta ratio in who are high anxious. And when you separately look at this wing of this particular design here, you can see that the decisions that you make also end up affecting theta beta ratio. So that is, theta beta reflects as changes in theta beta ratios based on instantaneous reward as well as cumulative reward, which we did notice was changing in these participants. So the only thing that is left to now check is whether changes that are brought about by trait anxiety to theta beta ratio, are they constraining the changes that the game requires in terms of mechanisms that enable the game itself. So for this, we actually did a mediation analysis where we looked at how how trait anxiety impacts theta beta ratio and how that impacts the game overall. And both of these arms were highly significant. That sort of clarified to us that in fact this dysfunction that you're seeing in the locus coeruleus ACC circuit could be indexed using theta beta ratio or pupil size. I'm not showing those results here. And those are being carried over from trait anxiety onto the game in terms of how suboptimally they perform. So this is what we did in experiment 3. So that is all great. I mean, this is still correlative evidence. This seems like there's something going on in this circuit, but we wanted to causally understand what was going on. For this, we actually causally manipulated the stress levels that these participants were undergoing. So what we did was we made them play the game once to get their baselines. We then put them through a very stressful stress induction protocol, hands in cold water that was at 4 degrees, very brutal. And then of course they were also interviewing for why they wanted a particular job. So they had to prepare a script and do this really quickly, and all of this when people didn't get any feedback. Sort of like, now when I can't see the audience, can't get any feedback, it is actually adding on to my usual stress levels during a talk, right? So all of this are potent manipulations that increase your stress levels, increase your anxiety, and then we made them play the game again. And the idea was that acute stress increases bottom-up salience. So it basically highlights what is happening in the environment, because you have to pay attention to what is happening now. And this salience here could serve as salience to the reward that are at your disposal. So trees that you're in could now appear more rewarding, and maybe that increases your engagement levels. And if the circuit is engaged and engaged to increase, circuit is engaged to increase engagement, then you might see that some of the effects that we saw earlier are alleviated. And this is exactly what we see. So first, of course, the manipulation is very potent. So you could see stress levels go up in these participants significantly. And then, of course, the slopes increase. So this is the slope which reflects how engaged you are in the patch. So if this is the regular, you increase your slope, meaning you're staying more engaged in the patch despite high anxious, high trait anxious conditions. So summary at this point is that we've developed a task that is very naturalistic, that taps on what we are thinking is your tendency to disengage because of hypervigilance. We've built a process model that explains the data and gives us some kind of quantification for this engagement. And this seems to be reflected by their increase in trait anxiety. And this dysregulation is coming about by this LCNE system, which is captured by these biomarkers and can be causally manipulated by acute stress, albeit temporarily. So this is all great. So I talked about this first part here. So now I'm going to talk about what this really facilitates. So this disengagement is actually in service of you're wanting to learn or understand what is happening around you. So how is this happening? Can we quantify that as well? So how does anxiety promote learning, and why is that good or bad? So this is what we're going to see. So learning, when I say learning, what I really mean is that any kind of learning in any kind of situation. So for example, it could be something like your visit to a restaurant that you're very familiar with, and you order the same pasta that you've always ordered. But today, the pasta is not tasting good. So this could be because of a true change. That is, the chef has now changed. So this is sometimes referred to as volatility, which is a true change. Whereas sometimes maybe it's the same chef, so there's nothing really happening. But because there are a lot of ingredients being used, there could be random fluctuations in the taste of the pizza or the pasta, and therefore maybe you should not make too much of the fluctuation and continue going to the restaurant. Whereas if it's a true change, you might actually want to make a significant update by signaling that you don't like this restaurant, or some kind of a belief update that makes you want to move away. Right? So essentially, a steep learning means a faster, quicker, more significant belief update, whereas a slower learning here means that you're sort of letting go and sort of continuing on with the same situation. So essentially, what has been known based on earlier studies is that people with anxiety misestimate this volatility. They assume things have changed truly when there have been random fluctuations, and this has been seen based on how they adjust in different environments. So to actually capture this and quantify this, we designed a slightly more dynamic version of the foraging task, so where things are changing over time, because our earlier version was actually something that was very static. So in this, people are actually just fishing by going to these 3 different islands, and if they get fish, then that's good, they get more points, whereas sometimes they just get worms and it ends up becoming a sad emoji or losing points for them. So essentially, over a period of time, they just have to assess and ascertain the best island.
Your time is up.
My time is up? Okay. So maybe I will just take a couple of minutes to run through this, and then can I take about like 5 minutes? Is that okay?
Yeah, can you just do it fast, about 2 minutes, please, because we're running short of time.
Okay. So I'll do it very quickly. So essentially, there are true changes. There are like changes that are just fluctuations. What we essentially saw was that those with anxiety, clearly as anxiety went up, they were becoming poorer and poorer in these highly stochastic environments that were due to random fluctuations. And what was really interesting, to cut it like really short, is that they were actually learning much faster. So all fluctuations were being treated as true changes, and they were learning really fast, and this high learning rate was actually was their undoing in some sense. So essentially, even though it seems like it's good to have a high reward learning rate, it's not good in noisy environments where things are, when there's a lot of fluctuation, because you're paying attention to something that is happening by chance, instead of paying attention to something that is stable over the long run. So, so that's what's happening in these conditions. They're learning too fast. In the last part of the study, I'll cover this in like the last minute, we wanted to look at sleep, because we know that this modulates anxiety levels, and can this actually also change the way people are learning in these conditions? There are early studies where people have shown that the prefrontal cortex activity is restored based on N3 sleep particularly, and this seems to reduce morning anxiety. So we observed that as well. So participants actually played the game, slept, woke up, played the game again, and we noticed how time spent in N3 sleep reduced their anxiety levels, restored their reward seeking in the morning, and brought it back to normalcy in the morning. So this was serving like some sort of a causal manipulation, the duration of N3 sleep, to restore impaired learning in people with anxiety. So essentially, to conclude, what I'm trying to say with all this is that pre-anxious people disengage from tasks easily, but increasing task salience reduces this tendency. They disengage because they're primed to learn quickly with minimal information, but sleep seems to reduce this tendency. And all of this is implicating the LCNE tone, which increases ACC activity, but it waits to be seen whether N3 sleep can actually moderate this by increasing top-down control. So with that, I'll stop here. I want to highlight people who've done all the work, the two at the front here. So Piyusha did experiments 3 and 4, Rakshita did experiments 5 and 6, and of course I want to thank all the funding agencies that enabled this. Thank you so much, and apologize for stepping over the time limit, and of course I shouldn't end without thanking Professor Venkat, Professor Bellare, Professor Mishra, and Professor Guwahati for inviting me to this auspicious symposium. And I'd love to have been there, but due to some personal situation, I couldn't make it. But hopefully I'll meet you all very soon.
So thank you very much, Dr. Arjun. So what are the clinical implications of his research? Uh, so during my residency about 30 years ago, we were only interested in three types of nervous systems. One was the central nervous system. The second was the peripheral nervous system, where we were looking at the nerves and all that. And the third was the autonomic nervous system, where heart and respiration, which you cannot control. But there is another very important nervous system which actually controls everything, which is known as the limbic nervous system. So now we find that emotion and anxiety and stress also influences every other aspect, which is the limbic system. And Dr. Arjun has been focused on this system. So what are the implications of this research? The first is, you can actually train and get trained in order to decrease your anxiety. So it's not like that you are an anxious person. It's just that you require some exposure and training, and with that exposure and training, it is possible for you to become less anxious in a particular environment. That's the first thing which can be manipulated. The second important thing is that, what exactly is experience? As a surgeon, when somebody asks me what exactly is experience, it's actually a series of bad experiences. So when you say there's a person who's a doctor who's a lot of experience, he's had a lot of bad experiences, which actually tells him what not to do. We do it for our children. Hey, don't touch that. That's fire. It's going to burn you. So this is a very important aspect of limbic system behavior, which needs to be manipulated. The third very important thing is controlling your emotional response to change. There will be two types of people, one who like travel, one who don't like travel. People who want to change, people who don't want to change. And this also can be actually manipulated by this important thing. Then the fourth is a very important aspect. Anxiety is not always bad. You have, how many of you, I just want a question from the audience, study on the last minute for an exam? I'll raise my hand. Who else? Okay. So what are we doing? What are we doing? What we are doing is we are getting ourselves into a state of anxiety. And that anxiety propels us to a state where learning becomes easier. And gradually we become so adapted to that we will intentionally postpone this to the last minute. When we can do it 1 month ago, we'll postpone it till the last minute so that our performance enhances and then we can actually learn. So this is a very important aspect. It is done by all athletes, Olympic athletes. They want to be anxious so that they can improve their performance. Then there is another aspect. Now we are talking about space, human space flight mission and all that. When we are actually selecting people for the human space flight mission, we are collaborating with ISRO from Sri Chitra. What we have to do is to understand that which type of personality that person is in. In a situation of stress, does he become anxious and deteriorates? Or in a position of stress, does he improve his performance? So this is something, so for selection of people, all these tests are extremely important. And the final point, which probably none of you have noticed, relates to this. It's not related to anxiety, but it's related to pupillary dilatation, electroencephalographic changes. How many of you are wearing a digital watch here in this audience? How many are wearing a digital watch? So what happens is, everybody, how many of you have a mobile phone? Everyone. Now the point is, wherever you are going, whatever you are doing, somebody knows, okay, where you are sitting, where you are going, what you're doing, what you're breathing, what your respiration is. And now when you're entering a shop, even your pupillary dilatation tells them that you like a particular thing or you don't like a particular thing. Say if you like a red purse, there'll be a little bit of pupillary dilatation, there'll be a little blood pressure increase, there'll be a little tachycardia, which the algorithms can actually understand. So like you get when you're surfing the net, you're continuously getting ads out of a particular time if you have clicked on one. Soon a situation will come that in all these sites, in all these selling sites, based on your limbic system response, you will keep getting those signals, which actually propels you to buy something based on that. We can't help it. We can't help it. But what we have to do is to understand it and master it. So I think studies like what Dr. Arjun has stated, we all need to work on this, and there are several aspects of this which we need to get into. So this is open for questions, please, and comments. Yeah, please. Okay, one or two questions, some important questions here. Yeah?
Sir, you mentioned that. What about the blood-brain barrier? Hello? Audible?
Dr. Arjun, can you hear?
Yeah, I can hear you, yes.
No. Ma'am, please go, be a little louder.
There is a mention that in your lecture it was mentioned that brain size increasing, recent research. Which part of the brain, whether the whole, because hypothalamus is the one which is directly affected by the factors, environmental factors.
Oh, I think you're probably referring to the slide where I said that foraging complexity has correlated with brain size. So here actually they're looking at the whole, they're looking at the skull size actually as a measure, as a proxy for how the skull size is changing with the foraging complexity. So there is, I mean, that said, I mean, I would say that there are...
1 minute. Skull size doesn't matter because brain size increases, folds increase, naturally folds increases.
Oh, no, I completely agree with you. But what I'm saying, I'm referring to the particular study that I was talking about. So in that study, so they are talking about how the skull size changes across species. And what you're saying is totally true. Of course, the folds increase the surface area and there are more cells packed in. And you also brought up the fact that hypothalamus is very, very relevant to this. But, are we also looking at changes in the prefrontal cortex across these species and how that matters to these changes? I mean, I think, it's a good question and a complex question. We don't completely understand how these changes are happening. But the only thing that I can say at this point is that all these signals from so for example, cortisol that is being released from the hypothalamus. So that is also...
Cortisol is not released from the hypothalamus. It is released from the adrenal.
Right. I mean, sorry, it comes back into the system, right? So it comes back into the system, and there are receptors. I mean, it's been triggered by the hypothalamus, but it comes back into the system, to the brain. And there are receptors, like glucocorticoid receptors, that are also highly densely present in the ACC. For example, ACC areas 25-24. So, I mean, the reason why I'm saying this is that even though some of these changes are being triggered by the hypothalamus, they end up coming and affecting other brain regions that are expanding, having more receptors for these.
I'll just clarify. So in the limbic system, there is a PAPAS circuit, which includes the thalamus, hypothalamus, the corpus callosum, the inducium gryzium, locus ceruleus. So it's a complete circuitry which is responsible for emotional reaction as well as memory. So all of them come together. Yeah. So
My
thank you.
question is actually how the blood-brain barriers affects. Skull size is not related to brain size.
Yeah. Ma'am, here the question of, I'll just answer that. The question of blood-brain barrier doesn't arise here because locus ceruleus is a very intrinsic part of the brain. So it's within the brain, which is...
Yes, sir, very true. But there are different types of nucleus in the hypothalamic part and where they respond only.
Yes, of course. No, so hypothalamus is responsible for... No, I'll tell you, the limbic system, like for example, the hippocampus has many aspects. One is hippocampus, amygdala. Amygdala is responsible for memory. Hippocampus is responsible for emotional response. And hypothalamus is responsible for the neuroendocrinal stimulation to whatever we are doing.
The first one to affect from the environment.
So it's a combination of everything.
Yes,
Thank you. We can conclude this session. So thank you, Dr. Arjun. Very nice presentation. Thank you so much.
Thank you very much.
So, thank you. Last but not the least, we are all afraid of growing old. And as we grow older, there are so many insecurities: financial, digital, social, isolation, health issues, insurance, so many issues come in. But there are some people who grow old even when they are not old. And these people are having Alzheimer's disease. And what is this Alzheimer's disease? You start losing your memory. And why is it so important? Because we are what we are because of our memory. We remember our near ones, our dear ones, our house, everything we do, our what based on our memory. And the biggest is that our own identity, our own world is our memory. If somebody asks me, who am I? The only way I can define myself is based on all the memories which I actually have. And there is a group of patients who prematurely lose this memory, and for them it becomes a huge issue. They start losing first their names, then they will start losing their areas around. And the old memories are the last to disappear. But soon they will even forget their children's name, their parents' name, and then that becomes a big issue. And there are many diseases which are responsible for it. In elderly age group, lack of blood supply to the brain is known as multi-infarct dementia. That causes disease, memory loss, which could be termed. There could be viral infections and prion infections which can cause Alzheimer's disease. This could also be because of one important disease which is known as Creutzfeldt-Jakob disease, which is a prion disease which can actually result in memory loss. Sometimes depression itself and emotional response can result in severe memory loss. And like you had Muhammad Ali, who received so many punches on his head, and he developed Parkinson's disease, and late stages of these degenerative diseases can also result in severe dementia, memory loss, and you can say, so nobody knows what the exact cause of Alzheimer's disease is, and this is one of the most important problems which is confronting India. Right now, India has a young population, but soon after 10 years, it will become an elderly population. And this is one area that we really need to focus upon. And it is my honor and privilege to call Professor Vipin Ramakrishnan, who's professor of the Department of Biosciences and Bioengineering at IIT Guwahati. He holds an MS in Applied Chemistry from Cochin University of Science and Technology and a PhD in Computational Biophysics from IIT Bombay. With over 70 international research papers, several book chapters and patents, his pioneering work in biophysics, bio-nanotechnology and network medicine focuses on molecular interactions and peptide-based therapeutics for neurodegenerative diseases like Alzheimer's. He is a fellow of the Royal College of Chemistry, Indian Chemical Society, Royal Society of Biology. Through his mentorship and outreach, Professor Ramakrishna continues to inspire young researchers and advance biomedical research in India. And he has a very interesting topic to talk about, and that is alternative therapeutic interventions for Alzheimer's disease.
Thank you, Professor Mishra. Thank you, Professor Mishra, Professor Venkat, and Professor Bellary. I'm very happy to, to present this work before you, and I'm very happy to have this panel also. Professor Venkat is a long-time friend. We were together, we were IYBA batchmates. And Professor Bellary was a very familiar face, which I see almost every other day when I was in IIT Bombay. So thank you so much for this opportunity to talk to you. And thank you, Professor Ladharangan, for this invitation. All right. So what I have to discuss is an unconventional therapeutic strategies for slowing AD progression. So the two speakers who have discussed their work before me were actually probably working in the area of neuroscience. But I'm mostly working in the realm of biophysics. So I will be speaking in that perspective. All right. All right, so because I think we have, it will take some time. I would like to be very unconventional because what I want to discuss about it in this particular discussion is about unconventional therapeutics. Is it working now? Okay. So I don't think I need to discuss about the hallmarks of AD, but the state of the art which I could find is that the factors such as neuroinflammation, AD pathogenesis is at this time is multifactorial and complex in its very nature. So we all know about Alzheimer's disease. Professor Misra has already given a brief idea about Alzheimer's AD in the introduction. Now, conventionally, when we started reading about Alzheimer's, these were the two hypotheses that which we were being introduced to. One is amyloid cascade hypothesis, and the other one is the tau ones. But in both these hypotheses, the basic research was focusing on A-beta clearance, on AB clearance. Now, in this particular work, what we are interested to discuss is mostly about the non-invasive therapies that can possibly decelerate the progress of Alzheimer's disease. So these are the non-invasive therapies that are available at this particular time in the literature, and probably some of them are in clinics, are scanning, ultrasound scanning, then electromagnetic simulations, light, and electric field. And this is from a review paper, and the last of them, that means the electric field, is actually our story. So we are getting into the story. So this is our subject of discussion. So electric field disruption of amyloid aggregation by potential non-invasive therapy for Alzheimer's disease. Now what we are going to discuss in this particular topic is that how an electric field can possibly decelerate the progress of AD. And we have developed two systems. One is an electric field based reaction setup, and other is a magnetic field based reaction setup in our laboratory for this particular set of experiments. Now there is a genesis for this particular work, and about 7-8 years ago there was this A-beta fibrillogenesis has basically about 3 nucleation sites. One is a dipeptide called FF or diphenylalanine, then a 6-7 amino acid peptide called KLVFFAE, and then a 42 amino acid long peptide, 1 to 42 A-beta fibrillogenesis, which we call A-beta peptide. Now we did a very random experiment, very out of a random curiosity, that what happens to this peptide or FF, diphenylalanine, in the presence of an electric field. So at that particular time in 2017, 2018, there were a lot of studies have been done on diphenylalanine. The entire discipline of bio-nanotechnology was grounded on FF, especially from the group of Yehud Gasset. So in this work of diphenylalanines, what we found is a very casual observation that it readily forms amyloid fibrils in normal conditions at 0 volts, but when we subjected this FF in magnetic field and in 2 different electric fields, 150 volt per centimeter and 300 volts per centimeter, interestingly we found no fibrils. So this is the genesis of this experiment. And this genesis of this experiment was actually not very random, because when I was a graduate student, I worked on a particular idea about the yin-yang hypothesis of protein conformational selection. The end result of that one was that the conformational selection in protein folding of a peptide or a protein is a result of a yin and yang of 2 opposing forces. The 2 opposing forces, one is a short-range electrostatic interactions and other is a long-range electrostatic interactions. And because these 2 countervailing forces are cumulatively acting on a singular system, the third factor, which is a sequence of a protein, is actually deciding the conformation of a protein. So this was the result, and it actually resulted in about 6 different publications on this very idea. So this, according to the yin-yang hypothesis, it is the electrostatics that drives the conformation. And if electrostatics is driving the conformation, then having FF being influenced or its conformation being influenced by an electric field is not random. There is a reason for that. What you see here is an amino acid, and a peptide is formed, is a polymer of amino acid, we know that. And in an amino acid, all around the surface, you will see charges. A helix, which is a polypeptide helix, we know that it has a definite dipole. This is something that which we learned in a classic biochemistry textbook in the very first chapter itself. If anything that has a dipole, when it moves, it will have an electric field. And if anything that has an electric field, then it should be influenced by another electric field. If you have 2 magnets, and if you bring another magnet very close to the other magnet, the first magnet will either come closer or will move away, because there is a field-field interaction. A field can interact with another field. So a field that is created by the dipole of a peptide should very well be interacting with an external electric field. So this is the core theme by which we construct this particular research upon. Now then, we have further made a series of studies by changing the stereochemistry of these peptide sequences, altering its electrostatics, and studied how it is differentially responding to various applied stimulus. Now then, what we did is that then we have understood that the first site of nucleation, which is a diphenylalanine, can be influenced by an electric field. We moved one step further. We took KLVFFAE, a 7 amino acid long sequence of A beta 1 to 42 peptide. And what we found is that under the influence of magnetic field, we could not find much of a difference. But in the presence of an electric field at 150 volt per centimeter and 300 volt per centimeter, the tendency to aggregate and form amyloids have decreased significantly. And this, what you see on the left, is a study with KLVFFAE. And when we found that it is changing in the KLVFFAE, we took the full 1 to 42 amino acid long peptide. We synthesized it in our laboratory. Partly we bought it from outside. And we did a series of 4 experiments. The last one is a magnetic field experiment, which we found no difference. But in the first one, we found that at 150 volt per centimeter, there is a significant decrease in the amyloid formation. And then in the second one, that is in the B of the right-hand side, what we did is that, we have the amyloid formation happening while the electric field is on. That means the conformational evolution is happening in the presence of an electric field. Now, in the second line of experiment, what we did is that we made preformed fibrils, already formed fibrils, which deposits in the cortex of the brain. And those fibrils we were subjected to electric field. And that is what you see it in the second line towards the right-hand side. And in the third one, experiment, we repeated this experiment in cerebrospinal fluid. Because in the first 2 experiments, we did it in phosphate buffer at pH 7.4. We repeated the same experiment in CSF. And we found almost identical results. On an average, there is a 35% decrease in the amyloid formation. So this is our observation. The same thing we could find it from our scanning electron micrographs and TEM micrographs. So what we could find is that this, rather than showing the data, I will just tell you what the end result of this one. We did how much it affects the neuronal cells. We took 3 different types of neuronal cells. And in those 3 different types of neuronal cells, the cytotoxicity or neuronal toxicity was found to be, on an average, about 17 to 21% less in the presence of an electric field. This is our observation. And when we made a very random calculation, it turned out to be extrapolating it at least 3-4 times, assuming that there are no other partial variables, we can offset the peak of the disease from 65 years to 79 years for an average patient. This is our observation. But at least if we could make it to 5 years or 10 years, we find it a very interesting observation. Now, again, when we further studied this, we go back to the drawing board, and what we find in the classic definition of Alzheimer's disease is that these normally soluble amyloid beta peptides misfold and aggregated into highly ordered insoluble fibrils. So this is the classic definition. So what we could find in this particular experiment is that in the presence of an electric field, the insoluble aggregates can become soluble ones, because when we can increase the solubility of these peptides, these amyloid fibrils, then it can have better chances of getting it excreted through the excretory mechanism of a body. So this is, so the next step is whether to have, this is some kind of a dream that which we have based on these particular observations, is whether we can have a wearable helmet, which we call it as an AD helmet. And this wearable helmet, if we can wear it for about 1 hour to 2 hours per day, then whether we can extend the peak age of the disease, whether we can offset it to about 5 to 10 years. And and I can show you if you want to. Now, okay, I'm keeping it here for the time being. Okay. Now, so this is the diagram. This is the circuit diagram, this particular work. And now, the first one, so we were trying to have 3 different kinds of interventions to a beta fibrillogenesis. The first one was the intervention based on physics. The second one is the intervention based on chemistry. And this intervention has a history also. And this history is the history of Trojan horse. Some of the students, I think most of the professors must be knowing about this history. So this is an incident that is kind of a myth that believed to have happened about 12th century BC. Because at that particular time, what happened is that there was a war between Greece and the city of Troy, because the Paris of Troy is believed to be kidnapped the wife of the ruler of Greece, and the Greece engaged into a 10-year-old war between the Greece and the city of Troy. And the city of Troy was very good in their military abilities, and city of Troy was working like a fortress, nobody can get in. So this is a trick that the Greek, because Greek were having some kind of a very important warriors of that time called Achilles, which you must be knowing, Achilles' heel and all. Achilles were in the Greece, and the Paris of Troy, and Curtain was on the other side. Now what they did is that they made a huge horse and left it outside the boundaries of the city. And this horse was having a quite large number of soldiers hidden in them. This is the mythology, don't ask me why, how, etc. Now, then these people from city of Troy found them, this horse is living outside, it's left outside. And then they brought it inside the city. And in the darkness of the night, the story is that these soldiers came out and eventually invading the city of Troy. So this is the story of city of Troy, and this horse is called Trojan horse. So in the chemical intervention, what we have done is that we have synthesized a series of molecules which can work as Trojans in a beta fibrillogenesis. So if you look at, so KLVF is the classic peptide sequence. And what we have done in this series of about 14 different peptides is that we have the same sequence change the stereochemistry of one amino acid. So it looks the same. It behaves like, it's like a spy in the opposition army. So we have about 14 spies, out of which 4 Trojans were found to be extremely successful. And the same technique we have used for tau proteins as well, where the central proteins which are prone to amyloid formation is VQI-VYK. We have made a series of peptides by changing the stereochemistry and not changing the sequence of the peptides, eventually intervening in the form. The work is that if you have these kind of amyloid formations and aggregations, if you have a mole in between, then it can create a wedge between subsequent fibrils in the protofibril formation, which eventually facilitate solvation of that particular peptide. And if it can move from solubles to insolubles to solubles, then most of the problems related to the position in the cerebral cortex can be alleviated. This is our theory. And this is the TAM micrographs of the same thing. And these are a series of, so in our device, this is a result of a series of simulations that which we did on the brain under the Professor Namade's and our idea was that if we are using electric field for therapeutic purposes, it should not get into the deeper areas of the brain. Instead, it should confine to the cortex of that one. So we did a series of simulations on the basis of this. And first, those simulations were on the mouse models. What you see is the 3D printing of the brain of the mouse model that is used for the study. The actual brain of the size will be 3 times more, 3x of this particular brain, because we need a model. I mean, we wanted to know the size of the brain in order to fabricate a device, because we have to do this first in brain. So this is how we wanted to restrict the field that is happening on the surface, on the cerebral cortex. So these are the people who have contributed to this study. The first row are the faculty members of this institute, and the second row are the students who have contributed to this study. So basically,
we employ a non-invasive regimen therapy for Alzheimer's
is the whole
using
purpose,
the electric
is the
field.
whole idea of this study. And this is our laboratory, and this is the MRI. Thank you so much for this opportunity. Thanks a lot.
Yeah, just one second. So thank you so much for these different methodologies. What you're doing is, now there are several aspects that you have covered. One is a chemical aspect, one is an electrostatic aspect, and one is a mechanical aspect, which mechanical along with electrical aspect. I mean, it's noteworthy. Only one small, there are a few points. We still don't know in medicine whether the neurofibrillary tangles and amyloidosis is the cause or the effect. Whether the Alzheimer's disease is because of the neurofibrillary tangles or it is the end result of the disease, we don't know that. So just manipulating the amyloid may not actually have the desired effect simply because we don't know whether it's cause of effect. That's the first thing.
I have mentioned in the state of that in the very first slide itself, the very same thing.
The second important aspect which I'd like you to, since you are doing work on amyloid, this research on Alzheimer's, I'd like to just mention one name. Someday she's going to win the Nobel Prize, but I'd like you to know her name. Her name is Maiken Nedergaard. Okay, Maiken Nedergaard. So what she did was, she's from the US, and what she did was, she discovered a very interesting system. We used to wonder when we were operating on the brain does not have lymphatic system. So how does it remove its excretory products? And neurofibrillary tangles could also be excretory products of the brain. And we used to wonder, why does a patient develop a lymphoma when there are no lymphocytes in the brain? And they don't come into the brain because of the blood-brain barrier. We used to wonder. What she did was, she discovered a lymphatic system in the brain which is known as the glial lymphatic system, for which she has won every single award except the Nobel Prize. And soon she's going to win that also. And what she discovered was that there is a glial lymphatic system which is working all around, and this specially becomes activated when a person is sleeping. So when a person is sleeping, has a good circadian rhythm, has long hours of sleep, then it completely takes away from the astrocytes and the neurons all these, these excretory products, which if it doesn't work, actually accumulate in the brain as various proteins, which could also manifest in hematoxylin and eosin stain like an amyloid. You understand? So this is a very important discovery, and which also tells us that when our elderly people used to say, please sleep well, it has a scientific basis, because the glial lymphatic system only works when a person is sleeping. It doesn't work when a person is active, and excretory system in the brain doesn't work. So that's one area that you need to focus upon. I think this is
are the ones which are most likely to cause disease, because fibrils are dead end. Soluble fibrils can bind to some other proteins and deplete the cell of the vital organs, and cell may die because of the soluble ligand. If you now encourage soluble fibrils, it might be counterproductive, one possibility. That's one just comment only. Second point, if you mentioned voltage, 150 or 300 volts, you said volts per centimeter. Yes. There's a gradient. Yes. If you get a field gradient of 150 or 300 volts per centimeter, your head being about 10 centimeters or so, the electric field voltage must be fairly high.
It's not actually so. That is.
Is it a field gradient or is it a field?
No, that is the whole. That is the whole work of their simulation. How can it bring down to the acceptable limits of voltage per centimeter?
Yeah, if it is, as you mentioned, 300 volts per centimeter, that's a fairly high gradient.
No, this is what it happens.
Yeah.
I'm sorry. Yeah. Okay. So basically, it's first of all, it's field. Field is not current. It's field. Okay. Second thing is that all our research, basically subsequent research, which we are engaged in now, which I have shown some simulations, are basically intended to fabricate the device which can bring down the field strength to acceptable limits. Number one, and number two, to bring down to focus the field in areas which are necessary. So right now we were working on the cortex alone, but since Professor Mishra was saying, we can localize it into this thing.
But also, I mean, many points like that. First is field gradient, and second thing is you're trying to simulate or use mathematical simulation models to say that the voltage surface coverage is there. If that is the case, then it doesn't do the job, because fibrils are inside the middle of the brain. So the field has to go to the middle of the brain, not only the surface. So that is, I think there's lots of engineering issues needs to be looked at. Yeah.
I think we have several questions, but I don't know. Yeah, maybe a couple quickly.
Yeah, I will be very quick. So what I'm asking, like, is there any dose dependence on this disaggregation process?
Dose dependence in terms
on
of
electric field or magnetic field?
Well, good question. Actually, no, magnetic field from our study, it doesn't work. It did not work. But electric field, yes. And the dose dependency, basically, well, it depends upon your molecular system. So in some molecular system, the amyloid formation or aggregation is decelerated best at 150 volt per centimeter. In some other system, it is best at 225. In other system, it's
just last and very quick. Yep. So, I mean, you are like disaggregating the real fibril formation, etc. Do you think it's going to inhibit the typical protein aggregation process, or you have already aggregated protein, let's say inclusion body, etc.? Would you be able to disaggregate using this kind of thing?
In our laboratory, we are making a series of materials which actually works on the modulation of these materials based on the electric fields. The answer is yes.
Is it working?
Yes. Yeah.
Thank you.
All right, thank you. I think we have several questions, but I'm getting instructions. But I think that's the last one. Okay, ma'am, you're the last one. Thank you.
So very nice talk by you, sir. So I have a couple of questions for you. Like one question is that does exposure to electric field also diminish tau fibrils? My second question is, did you also investigate the effect of electric field on amyloid beta peptides other than 1 to 42 peptide? Like amyloid beta 1 to 29, which is shorter, but much more toxic than amyloid beta 1 to 42. And my third question is that did you also do the in vitro study apart from this in vivo study that you conducted? First one is that does exposure to electric field also diminish tau fibrils?
I'll answer. Thank you for
Short
asking
answer,
the first
please.
question. The first one, we have not investigated that. Second one, we have not investigated 1 to 29. Instead, 3 molecular systems, FF, KLV, FFAE, and 1 to 42, and 1 to 40 are the 4 molecular systems which we have worked on. We have not worked on the 1 to 29. And what was the third question?
And so the third question is, did you also do the in vitro study? Like, did you use any amyloid beta model? Did you use any Alzheimer's model in vitro culture study, apart, like other than this one in vivo study?
No, not really. Because once we found that, our idea is to quickly get into kind of a therapeutic solution, if it is possible. Like, so we were so much focused on that one.
Okay. Thank you, sir. Very nice talk.
Yeah, thank you. Thank you very much. I know there are a few other questions, but I'm sorry we have to close. And it's been a great opening session. And I of course thank the speakers immensely for their valuable insights. I think we've covered a range of disciplines, and I also thank our chairperson to bring in the clinical insights and the cutting edge research insights into all the talks that have come. So thank you all. Thank you all. We close this session.
Can we have Professor Sanjay Bihari on stage to felicitate the speaker, please? Can I have Professor Vatsala Thirumalai on stage? I'd like to request Professor Sanjay Bihari to please felicitate Professor Vatsala Thirumalai. Can we have a big round of applause, please? Now I would like to call up on stage Professor Vivin Ramakrishnan. Professor Bihari, I would also, yes. Professor Bihari, I would request you to stay back on stage. Now can I have, please have Professor Jayesh Bellare to felicitate the chair of our session, Professor Sanjay Bihari. Can I have a big round of applause, please? Thank you, professors. We are closing the session 1 as of now, and we have the arrangement of the tea outside of this hall. We'll be meeting in the same hall for the session 2, sharp at 4:10 p.m. Expecting to have all of you us together.
So we are running quite late. So I would really request everybody to be back in 10 minutes, please. We have one more session to go. So please be back in 10 minutes.
start
Recording
around
in
now.
progress.
Yeah. No, I'll start with your introduction, sir. Good afternoon, everyone, and welcome to the second session of 95th NASI Annual Session and Symposium. We are privileged to welcome Professor Ashok Puranik, Executive Director of AIIMS Guwahati, as the Chair of Session 2 of 95th NASI Annual Session and Symposium. Professor Puranik is an internationally recognized trauma surgeon and surgical intensivist with over 3 decades of exemplary service in the Indian Army. His career spans major national operations, high-altitude medical care, disaster response and humanitarian missions, including Operation Pavan in Sri Lanka and Operation Vijay in Kargil. A postgraduate of AFMC, Pune, and a trauma fellow from the Alfred Hospital, Melbourne, he has been instrumental in advancing trauma systems, modern wound care techniques, and surgical training across country. He is a respected author, educator, and a strategic advisor in healthcare and technology. At AIIMS Guwahati, he envisions strengthening the institute's pillars of quality medical education, evidence-based patient care, and cutting-edge research. Sir, we warmly welcome you and are honored to have you as the chair of session 2 of this symposium. And I would also like to request the speakers to please confine to less than 20 minutes because of the shortage of time.
Good evening, everyone. I think in just one more line of introduction, this is my third tenure in Assam. As a captain, I was here 86, 88, and as a young surgeon in Nagaland, Manipur, where I learned art of managing gunshot wounds. And this is the third time I'm back to the Assam, where I started my career in a place called Changsari, and I'm back to Changsari. We are going to have a session with Dr. Neelam Sinha and Dr. Subhadeep Bhattacharjee. Neelam is Associate Professor with the Brain Research at IISc Bangalore, and she is got 100 plus research papers. She is working on advanced neuroimaging and artificial intelligence, especially on the aging of the brain. They are trying to detect the early cognitive decline and neurodegenerative disease especially. And she is a leader in the project of SANSCOG and TLSA. And she is a researcher, scientist, and innovator. And that will be followed by another young person, the young researcher and the engineer Subhadeep Bhattacharjee, who is assistant professor with the IIT Hyderabad. He is an electrical engineer, and he is also PhD in semiconductor devices, 2D material-based transistors and synaptic devices. But his research has got an award from the nuclear sciences, and he leads the nano device lab pioneering neuromorphic hardware for low power AI, and he is advancing the nanotechnology in India, especially in the intelligent hardware innovation. So without any ado, I hand over the mic and presentation to Neelam Sinha. If you could, because of short of time, as early as possible, if you could really present your views on this, that will be appreciated. Thank you. Neelam Sinha ji, over to you.
Thanks so much. I'm so sorry about not being there in person because of the Indigo fiasco. Is my PDF visible?
Yes, ma'am, you're audible.
No, can you see my document, my slides?
Yeah, it's visible. Neelam ji, it's visible.
Is the change of slide also visible?
Yeah, yeah.
Yeah, yeah.
Could you increase the volume a little bit this side, our system here?
So good evening all. I will not take much time. You've been listening to so many different perspectives. So this will be a very different kind of a topic. So here I want to look at how generative AI is being used in neurodegeneration studies. In our daily lives now, we see generative AI almost everywhere. everyday conversations, education, painting. For instance, here, you just have to give this prompt and lo and behold, there's a painting there that captures exactly the details of the prompt. Poetry, Shakespearean sonnet style, here you have a poem. You want to make a movie, so it's generative AI is in every aspect of our lives. And especially when it comes to chatbots, we are spoiled for choice. All of us are using this regularly. Perplexity is right on our mobile, ChatGPT, everything, right? So we are really spoiled for choice. But why don't we see so many choices when it comes to medical imaging? Now, what's making this possible in the other domain is innovations in algorithms, differences, the advancements in hardware, and availability of lots of data. So this is the most crucial thing. And the issue with medical imaging, as we all know, data is so sensitive, there are issues of privacy. Annotation is not for layman, it is only for expert. Very expensive to acquire. And the data that we acquire, they come with so many issues. Were the right settings used? Was there an emotion? There are so many issues that can crop up, right? Is the image adequate enough for diagnosis? Only then you can move ahead with it. Besides that, there are different challenges like what is normal? There's so much diversity in what is normal itself. And when you look at deceased, what stage of disease, the rate of progression, everything matters. Huge variations in hardware. So there are so many issues that make it very hard to give us consistent amount of data that is needed. But then the potential, the possibilities that we can have by using generative AI in medical imaging is immense. Denoising as a very innocuous example. Super resolution. I start with a 1.5 Tesla MRI. Can I end up with images of the quality of, say, 7 Tesla MRI? That can be so much cost saving. You can reach hard to reach areas also, right? Modality conversion. Can I have CT and go to MRI? Can I have MRI go to CT? Can this be possible? Can I acquire only T1 weighted MRI and end up with, let's say, a flare image, MP range? So can I look at different contrasts by just acquiring one contrast? Can we have anomaly detection which is automated? So the heat map is trying to show the region of anomaly. So the potential to generate meaningful synthetic data sets can be used in myriad ways. It can be used to predict diseases in a given time frame. I acquire images now. Can I say 2 years from now, given the other conditions, what the images are going to look like? And is it a predictor of a disease? It can help us with complex rare diseases. Subject-specific treatments can also be thought of. So this potential is huge. It must be tapped. We are already seeing some bit of this in action. I saw this website, I think at Yale University, where they have this ECG GPT. They ask you to upload ECG image, and then the model will tell you if it is healthy or unhealthy, and if it's unhealthy, where exactly is the problem? Which lead, which part of this time series is telling you that there is a problem? So it's already a step ahead. So one popular framework of generating synthetic medical images is MONAI. It's available for the last 4-5 years. Look at the kind of effort that has gone into it. All bigwigs, Stanford, King's College London, so many universities, hospitals, and then the muscle power of NVIDIA, all put together have been responsible for this. It allows one to train, evaluate, and deploy generative models on medical imaging. So I couldn't resist showing this, this snippet where it says, create infinite medical imaging AI from NVIDIA itself. There are lots of studies that are being reported now with synthetic data, and you see this across anatomies, across modalities. But this is for task-specific scenarios. They tell you, is it for segmentation? Is it for classification? Is it for lesion detection? It's all happening. Now, what is the principle of generative AI? We have lots of data points of a particular category. We do not know its distribution. So the whole goal is to try to learn this distribution. So we try to come up with a model distribution that we hope is mimicking the actual distribution, and then we pick up samples from this model distribution. That is the key idea of generative AI. How do we know how well we are doing? We could be doing visual comparisons. So here on top we are looking at the real images. At the bottom we are looking at the synthetic images. There's definitely a loss of details, right? So how do we measure the goodness of performance? There are lots of quantitative metrics that people have come up with. FID, SSIM, Structural Similarity Index, Maximum Mean Discrepancy, where you're looking at distances between the distributions or statistical features that are derived from the distributions. So these are some quantitative measures. SSIM tells you perceptually how close is the synthetic image to the real. So there are different ways of looking at how good the generative model is working. Some of the paradigms here, the very first ones were variational autoencoders, but then the more successful ones were GANs, followed by diffusion models, and the current one is called the flow models. So we will look at some examples from GANs and diffusion models. Now GAN is more than 10 years old, stands for Generative Adversarial Network. It was proposed on natural images, and it took a while to be adapted for medical images. So the first work on MRI images popped up only after 2 years. So how does this work? GAN has two big elements. G stands for the generator, D stands for the discriminator. So the idea is to show the generator noise and teach the generator how to create an image out of the noise. So X is the generated image. The discriminator D will compare the generated image X with a real image Y. As long as the discriminator cannot say whether X is fake or real, this training will go on. So when the discriminator can be fooled, you know you have trained enough and you will start using this to generate your synthetic images. So here what we are seeing it is on a specific case of the MR images. We are feeding random noise into the generator. There's so much learning happening. It creates a fake image. There is a training set of original real images. The two are compared by the discriminator. What does the discriminator say? Is it a real image or fake image? So when the discriminator can be fooled into calling the fake image real, we know our job is done. So it's a dynamic interplay between the generator and discriminator. The objective of the generator is to create samples that are indistinguishable from real data. So the goal is to produce realistic data points which are indistinguishable from the original data set. So this is the first work, one of the first few works in 2017, where they used GANs to convert MR images to CT. So when you have subjects who cannot be exposed to radiation, safer to do MRI and then look at the CT images. It took a while before realistic artificial brain MR images could be thought of. So this happened in 2018, where they used GANs for creation of MRI images. So here you have the image generator, the discriminator. The discriminator is telling us whether it's fake or is it real. So here you see a comparison of the generated images versus the real images. Now the first thing that you use the synthetic images for is to augment. The next thing you start doing is, can you analyze a disorder using those synthetically generated images? So one of the applications is to look at AD progression, Alzheimer's disease progression using GANs, which can be subject specific. So the goal is to predict changes with AD progression using subject specific model. So here synthetic images are produced based on a particular individual's MR images at different progression levels. So the model is trained to do both. It can introduce and also remove AD-like features from different regions of the brain. We'll quickly see some examples. So the top panel shows you the entire brain and the bottom panel shows you a zoomed version. The real image. Now, if we can quantify symptoms of AD, if we can capture them, it will be enlargement of ventricles, the widening between these, the fragmentation that we see, right? The widening here. So here, for a smaller measure of AD, you see smaller enlargement. For a larger measure of AD, you see much more larger enlargement and so on. So they are trying to quantify disease progression and convert it to the characteristics you would see on the images. Now, yet another way of doing it is not by altering the image itself, but by looking at what are called the latent space embeddings. So the image is given to the encoder, which creates feature embeddings. So now you have a compressed version of the image in what is called the latent space. You make changes or you understand the progression by looking at changes in latent space. So this is far more subtle than tampering the image itself. So what is the advantage? You have the person's acquisition right now. You know what the progression timeline looks like. You know how the changes will affect the image in the latent space. You make the same changes in the latent space and then you try to predict what the next acquisition is going to look like. So here you are generating images that mimic progression. So 2 years down the line, if you see that the actually acquired images look like what you have generated, so you know that the person is in danger. Another variation of GANs is called conditional GAN. Here, apart from noise that is fed to the generator, you also feed, you can feed a Boolean variable, or you can feed, let's say, some characteristic, age or genetic predisposition. You feed something more about the subject so that what images you generate, they are more reflective of the person. So the input image can be transformed into a different version of itself. So there can be a change of modality, style, or a constraint. So we'll see how people have used it. The input image can be transformed into a different version of itself. So this is what we said. So how are they using it here? You're generating images. Along with that, there is an attention mask that will decide how much weightage to give to what is generated. And I mean, we are looking at an addition of the two, where the areas that are affected are captured in this attention mask. So A is the pixel-wise attention mask. So the generator can focus on transforming those brain areas where the disorder will induce deformation. That's the goal. And you have to keep the other regions intact, right? So you do both. So that is the meaning of this addition. So you are deforming it on one side, the other side you are keeping it intact. The regions which are unaffected are kept intact. So finally what you get is a more realistic version of the disorder. So here's an example of how, with varying levels of severity, you see how the images change. The same signatures, enlargement of ventricles. So what we are seeing here is the attention mask. Changes in the attention mask is leading to changes in the generated image. Now, apart from GANs, there is a very popular model called the diffusion model, which is very successfully used in computer vision. So here just like in the previous case, the goal is to generate synthetic images, but here it's done in an iterative way. There it was a one-step process. In the forward process, you start with a good, clean image and iteratively add noise till what you get is just noise. Now, why have we done this? We've done this so that we can learn how to denoise. So the forward arrow is important here. So we did this so that we can learn how to go from noise to the structured image. So the forward process, that is simple and fixed, but the reverse process, that is where the whole learning happens. So we are, at every time step, we are creating a noisier version of the original, that's in the forward process. In the reverse process, you learn a network to approximate the reverse distribution. So at the end of this, you should be learning what was the noise you added, and more importantly, what was the clean data. Like every model, the first thing that's done is to create data for data augmentation, because we know any of these deep learning based approaches requires lots of data. So can we augment data? This is a very recent work, diffusion models for neuroimaging data augmentation, assessing realism and clinical relevance. How else has it been used? To understand disease progression. Very similar to what we saw with GANs. This is remarkable. There's a 70-year-old who's been followed for 15 years. They are comparing the actual acquisitions with what they could generate. And what you see in the top panel is the difference. The difference is so small. It means that the images they have generated has been able to match the ground truth perfectly. But this comes with a lot of input. They used lots of images, upwards of 11,000 from several publicly available data sets. It has to be age matched. The data set has to be very well balanced. And then you see the metrics, the mean square error. Look at this longitudinal, the last two columns. The MSE, mean square error, is 0.004, so which is a very, very good achievement, right? And even SSIM of 0.91 is a very big achievement. We know SSIM varies from 0 to 1, 1 being the ideal. So once you cross the 90% mark, you're almost there. But as we said, it takes a lot of resources. But how practically useful are these generated MR images? So visually, you can see there's a lot missing. So it's important to formulate an exclusive quality index for synthetic MR images for a given condition. So GANs, though we saw very good results, we know they are prone to hallucination. That is, they generate details that are not faithful at all. And diffusion models, they give us better quantitative metrics, but they require more input data and they are far slower. So in a nutshell, there's so much possible. You can define the condition, you can think of better ways to reconstruct, you can think of changing modalities, you can think of changing contrasts. So there are several applications of Gen AI in neuroimaging. But then there are several open problems that need to be addressed. We need to find ways to integrate domain knowledge, clinical assessments. We need to find ways to integrate patient history, comorbidities, genetic predispositions. But most of all, we need MRI-specific, brain-specific quality measures. The current ones were all devised for natural world images. So we are just applying it blindly. So we need task-specific brain image images. Thank you. This is all I had to say. I'll be very happy to take any questions.
Thank you, Neelamji, for updating us about generative artificial intelligence and how you're working on MRI pictures.
Thank you.
Okay, next we have Subhadeep Bhattacharjee. He's an electrical engineer from IIT Hyderabad, and his forte is research in nuclear sciences, especially on a nano device lab pioneering neuromorphic hardware for low power AI. So over to you, Dr. Subhadeep.
Hello, are you able to hear me?
Yeah, yeah, you are very much audible.
Okay, just share my slides.
Okay, you have 15 minutes with you. Okay, thank you.
Are you able to see my slides?
Yes, yes.
Okay, just, yeah, you're able to see my slides, right?
Yep, yep.
Okay, thank you. So thank you very much for the warm invitation. Again, I'm sorry not to be able to make it. Um, as introduced, I am basically a nanotechnologist, more specifically a device physicist. So we work with electronic devices. And today, instead of talking about my work, I would try to motivate how nanotechnology is now being employed to solve some of the very deep issues or deep trouble with deep neural networks. We know a lot of success behind the deep neural networks, but it comes at a huge cost. And that cost, to understand what that cost is, we will need a more hardware perspective. So, very brief outline, we will just motivate what is AI/ML computing, why do we need AI/ML computing, and how the first generation of AI/ML computing came from inspiration from biological neurons and synapses. And that is what creates a lot of problems, because there is a fundamental mismatch between what is happening in software and the current computational systems. They were never built to run AI/ML algorithms. And therefore, we will talk about hardware inspired from the brain. And that is the area of neuromorphic computing and devices. We will give a very brief um, introduction to it, and maybe 1 minute of what we do in our work. Because in a very generic audience, talking a lot about my work may not, motivate a lot of questions. So one thing that we need to understand, especially because we all are interested in computing, is that we need to note that there has been a radical and a dynamic shift in the way computing has been has been done since the last 30 to 40 years. If you remember, just 30 years back or 20 years back, if you go to a high performance computing conference, most of it is focused on how do you solve computational fluid dynamics, basically solving differential equations to give you deterministic results. And that was your compute centric paradigm, which got completely changed to what we see now and we know as the data centric paradigm, where what you're doing is essentially heuristic computing. What is heuristic computing? You take a huge chunk of data, and there are no right answers, but what you're trying to essentially do is you try to recognize useful patterns and classes from rather unstructured data. And the more important thing to now bring to the fore is what has driven this change. And I would like to argue that nanotechnology has really driven this change. The availability of big data in form of advances in both memory and storage technology, and the huge amount of computational firepower that nanotechnology and this MOSFET scaling has brought to the fore. The next thing that we want to kind of understand is, why is there so much attention to AI/ML computing? AI/ML computing very primarily is because a long time back people understood for doing certain computational tasks. I'm not saying all computational tasks, not simple number crunching. A baby's brain is much more efficient, both in terms of power and performance, compared to even today's best supercomputers, and that we will see. And what are these tasks? As I mentioned, these are tasks of pattern recognition, classification, decision making. Of course, arithmetic, number crunching, finding the logarithm of a number, or doing prime number factorization. Still, there are these big, powerful supercomputers. But if you have to find patterns out of some unstructured data, even a baby's brain, just by seeing your face in 3 or 4 times, it is able to understand that the baby is able to understand this is a friendly person or an unfriendly person, despite a lot of contextual noise that surrounds the so-called interaction, right? So I will not go into details of what AI, ML, and deep learning is. Simple AI, artificial intelligence, is a big umbrella term. Machine learning is one way of including artificial intelligence, and this is where things change. Instead of your classical computer engineering, where you write a series of handwritten codes, in machine learning you learn from data, you try to learn patterns from data, and then you try to do inference. And of course, deep learning is doing machine learning with artificial neural networks. Now, without going into details, I just want to break the myth that AIML and this paradigm is a new paradigm. It is not. Actually, the golden period of AIML actually was in the 1940s to 70s, where all these big ideas, whether it's artificial neurons, backpropagation, the Turing test, perceptron models, all were really available. And the theoretical underpinnings and foundations were already there. But it took almost 3 to 4 decades for AIML to really take off. And that is the first question that we will ask. Why was this huge lull period of nearly 3 to 4 decades? Why was there a big gap between ideas and execution? And what suddenly changed after 2000 such that you had this big boom of AI? And that also makes us ask a very important question, which is going to decide what we have to do in the future. Like I said, if it has run out of steam earlier, all these big ideas have run out of steam earlier, can it happen again? Can AIML run out of steam? I would argue, if we are not careful what we are doing at the hardware perspective, it will run out of steam, and that is the story that I want to kind of share today. So maybe this is not really essential for trained scientists, but for students, I try to always explain that the fundamental, if you ask what is the fundamental task at hand at AIML, it is essentially conventional curve fitting. As experimentalists, we know we do an experiment, we get this huge scatter of plots, and your origin or any simple Python code also can do conventional curve fitting, where it guesses the polynomial and then computes the cost function. How far is my prediction away from the actual data? And by minimizing this continuously and iteratively, it will be able to fit data through this scatter of plot. This is exactly what is happening in AIML as well. You are essentially trying to do curve fitting, but now at a hyper-dimensional scale. You just don't have two-dimensional data. You have several dimensions in data, as you can see from this very simple AIML task of handwritten digits. So a pixel, the number of pixels is one dimension. What is the intensity of pixel is another dimension, so on and so forth. So the main question or challenge in AIML is, how do you build a generic framework which can do hyperdimensional curve fitting? So that is the key problem at hand. And since the brain does such a great work at doing hyperdimensional curve fitting at relatively noisy environments and still does it at such low power consumption, the inspiration right from the 1940s was to try to take inspiration from the brain. And as you know, I don't need to explain this to this august audience. You have a huge interconnected web of neurons in your brain, and these brains communicate, and these neurons communicate.
Dr. Shubdev, you have 5 minutes.
Yes. Hello?
You have 5 more minutes.
No, carry on. We have till 5:15.
No, I don't think I have even finished.
Yeah. Okay, carry on. Subhadeep, carry on.
Yeah. So you have brain. I don't think I have finished even 7-8 minutes now. So yeah. Anyways, so so as we know, brain is an interconnected web of neurons, and this interconnected web of neurons, at the junction is where all the magic happens. This is your synapse, and the synapse is what decides the strength or the weight between these connection between the action potential and what effect it happens. It results in the postsynaptic neuron. And this is what was the inspiration behind the software neuron, without going into details. Just like the fundamental structure of these neurons and synapses at the brain, you mimic this at the software level, where you have linear and non-linear components, where the linear component is your multiply and accumulate operations, whereas your non-linear operation is your activation function. And this is how, by scaling one software neuron over different layers and increasing the depth of the layers or the number of hidden layers, we come to these very powerful deep neural networks. And that is what is able to do all the magical things that we see around, including what our previous speaker talked about generative AI. The fundamental structure is actually inspired from the human brain. So without going into too many details, how do you train these neural networks? Just like how you train a baby, You point at an object, apple, orange, or whatever it is, several times. You give it certain labels, and you say that this is what the object is. And the expectation is your so-called backpropagation algorithms will be able to adjust the weights inside the neural network such that you will now be able to do a efficient inference or make predictions. So in a very short time, maybe I have been able to explain how, taking inspiration from brain, you are actually able to make artificial neural networks train and also do inference. But the problem now comes where the problem. So, how much computational resources do these dragons, as I like to call it, because they are extremely powerful machines, but they are also very, very resource hungry. And this is where all the problem starts. They are extremely resource hungry. We know by the fact, without getting into too many technical details, we know that to just train the deep learning project of Google on basics, just 6 tasks, just 6 features, flower, pizza, or whatever it is, something that human beings can do in a matter of seconds. It took 3 days just for it to train the system with your Google supercomputing system. Not just that, the amount of computational firepower that you nowadays need to do even simple tasks is really off the roof. And the main problem comes when you start looking at the power consumption numbers. And this is where the reason between the ideas and the execution. While the ideas of AIML computing were really formed by the late 60s, it took lot of computational firepower, which we now have in our hand to do AIML computing. And this ability from nanotechnologists to scale the transistor and to include many more transistors in a small footprint is what is leading to your NVIDIA's GPUs or your Google's TPUs. So I think a hat tip for the nanotechnologists and the semiconductor device community is absolutely essential in understanding what made us realize this dream. Okay, but not only that, that it takes a lot of computational firepower. The amount of power consumption with this dragon is not only very hungry, it's a very angry dragon, because your simple task of just finding 6 features or 6 images takes thousands of kilowatts of power. Now, if you look at in the context of Chat GPT, your GPT-5 model that we keep using, the number of days of training it required was almost a year worth of training from the internet. Look at the power consumption. It is 0.15 billion kilowatts of power. And that can almost power 25,000 Indian households per year. What was very shocking that I read very recently is the current data center power consumption is more than that of France, and by 2030, data centers of the world which run these AIML algorithms will consume more power than even India. So a natural question that comes to the picture is, why is this huge power consumption between when we try to run these AIML algorithms on even the most sophisticated hardware that human beings have built? And the answer, the very simple answer, is there in this particular slide. What happens is the way our computational models have been built for the last probably 6 to 7 decades, and it has not changed, is very counterintuitive to the actual hardware infrastructure of the brain. In brain, as you saw, there was no difference between what is logic and what is memory. The neurons and synapses and a web of neurons and synapses form the entire infrastructure of logic and memory. Unfortunately, the von Neumann architecture, on based of which all of our computational systems are working, is not based on this philosophy. As you can see, logic, where you do your computation, and memory, where you store the values of computation, are physically and functionally separate. So what happens is the data shuttling back and forth from the logic to the memory elements, that is what causes all the latency, and that is what causes all the power consumption. So as you can see from this graph, one multiply and accumulate operation costs almost 200 or 500 times lower than the data it requires to shuttle or the latency and the power it takes to shuttle data back and forth from the logic and memory elements. And this is the main inspiration of working on neuromorphic computing devices. What is the idea of neuromorphic computing devices? It is the recognition of the fact that current TPUs are absolutely unsustainable in terms of power consumption and the impact they have on nature, the in terms of both performance and power consumption, and looking at the fundamental incompatibilities that AI/ML software has with current hardware infrastructure. So the idea is, and this is the area that we work on, is this very fascinating concept of neuromorphic computing. So what we essentially do in neuromorphic computing is we start taking inspiration. We realize that just taking software inspiration from the brain is not enough. You need to have a clear understanding of the hardware infrastructure of the brain as well. As I explained, while the current one Neumann architecture has separate memory and logic elements, your bio-inspired computing, everything is collated. And that is why the promise is that while the modern day systems consume huge amount of power, your brain, which is working at fraction of the power, will be able to do all these things because there is no continuous shuttling of data back and forth. And these are the kind of computing systems that I and all many other people around the world are trying to build. The place that I focus, without going into details, is on the memory aspects or in the electronic neuron aspect. So we have made software neurons and software synapses. They are what consume this huge amount of power. So why can't we leverage on all these great advancements from semiconductor technology to build artificial neurons and also artificial synapses? These artificial neurons and artificial synapses, the promise is that they will not exactly behave or they will not exactly mimic all the biophysical mechanisms of the neurons and synapses, but only that much which is necessary to lower power consumption in modern day computing systems. And without going into too many details, what this is the work that we focus on in our lab. We try to work on new age materials for neuromorphic devices and for neuromorphic computing. So there are some works that we have published where we have looked at the thinnest resistive RAM device, which is behaving like an artificial synapse for artificial neural networks. But also we look at charge trapping and detrapping devices at molysulfide dielectric interfaces, which behave like synapses or spiking neural network. And these are able to mimic the fundamental uh, learning rules that are there in your artificial synapses. Some of you may be familiar with it. They are called spike timing dependent plasticity, pulse bed facilitation. The fascinating area that we work in is how do you mimic these biophysical mechanisms using electronic devices which can now be made in a which can be scaled in a small chip and then be integrated in a full scale hardware. So this is what we work on in our lab nowadays. We not only work primarily on 2D materials, we have also scaled our understanding of things from looking at just device level data to start building systems. And we see indeed that one of the key advantages that these neuromorphic devices give over traditional ways of computing or neuromorph or one Neumann way of computing is that not just they are much low power, they are also much resilient to noise. But of course, it is coming at a price of a little bit more latency. So not just this, with my collaborators in chemistry, we are also building uh, biocompatible ferroelectric molecules which are compatible even as implant so that we can extend the idea of neuromorphic computing not just in a semiconductor chip but probably even in implantable devices. So this is what we kind of work on. The concluding thing that I would like to say is that uh, the final message would be that the rate at which we are burning power by which is due to the incompatibility of AI software and current modern hardware, is simply unsustainable. And that is causing this huge amount of power consumption and latency. And we need a lot of thinking about whether this is sustainable, and a lot of research investment in finding ways to do edge computing for it. Okay? So if you find this area very interesting, I have an upcoming NPTEL course which discusses all these things in detail, not just memory device technology, which ushered in the AIM revolution, but how these memory devices are now being augmented for neuromorphic computing. So the enrollment is open for any students who are attending this, and I will like to end by acknowledging all my students, my collaborators, and the funding agencies. I'm happy to take questions if we have time, otherwise. But thank you very much for this nice opportunity.
Thank you, Dr. Subhadeep Bhattacharya. If audience has any question related to the nanotechnology and its integration in the hardware. Any questions? Yeah. You can write also on a message. I think there is no question. Any question for Neelam Sinha related to the artificial intelligence, neuroimaging? Okay, Subhadeep, I have one question. Compared to the Western science lab and scientists, where do we stand in terms of our contribution, our innovation in novelty?
That's a wonderful question. Thank you very much. So I think where we are doing almost equal to what is happening according to Western science, especially in the area of neuromorphic devices, is only at the device level. So we have a lot of intelligent people who are working on new concepts of devices, but where we are really lacking behind is in the system level integration and system level implementation to realize systems that can be run in the chip. But I would like to mention here Professor Sritosh Goswami, one of our colleagues from ISC Bangalore, from SENS. He is doing wonderful work. He has a lot of now couple of papers in Nature itself, where he has taken these organic molecules, which he has been working from in his PhD thesis, and actually showed made systems and run hardware algorithms to show there is a distinct advantage of using this kind of computing. So kudos to him. And I think we are all catching up to him and other people in the world. So we are doing very well in terms of devices, but the system level integration is where we need to catch up.
Thank you, Subhadeep. I think today is the time when data science and artificial intelligence and nanotechnology has to be a common subject for all, right from the beginning. Okay, please.
Absolutely, sir.
Subhadeep, we have one question. Okay.
Yeah, Dr. Subhadeep, for fascinating presentation. See, you were talking about how to optimize computing. I'm just looking at the other perspective, like using the neuromorphic computing models. Can we understand the brain better so that we can make it healthy and understand why it gets disordered?
What a wonderful question. So your idea has been implemented by two main players. One is in Japan, I don't remember now the research lab, and one by IBM. It was called the Brain Scale Project, if I'm not mistaken, where they using computers simulations and GPUs and TPUs. They are trying to mimic a fraction of the brain's computational power. Unfortunately, I mean, we are way behind nature. So what they showed was to mimic just 1% of the brain's interconnectedness in form of neurons. It took almost 5 megawatts of power. And I don't know how many $1 million they spent just to mimic just 1% of brain's activity. And the reason for that, the fundamental reason that of that, probably electrical engineers will appreciate it more, is something called fan out. The meaning of fan out is fan out. The meaning of fan out is how many neurons is then connected, what is the interconnectedness of neurons. In the brain, it is 1 to 10,000. One neuron is connected to 10,000 other neurons. In the best of CMOS circuits, even to make a fan out of 1 is to 100 is a very, very, very big challenge. So if we cannot crack this problem, I think we will still be very far away from the brain. And the ability to try to mimic, that's why I said neuromorphic devices, we never try to mimic the brain exactly, although it may have certain applications in terms of Alzheimer's and so on and so forth. But that is not the objective, because it's very difficult for us to gain this fan out. How do you have this large interconnectedness, which our brain is able to do? That is some fascinating thing which really stumps us. So maybe I hope I was able to give a flavor or answer the question.
Yeah, exactly. So the brain is so complex that there is billions of neurons and then billions of synapses, and then it connects to the nodes, the various big junctions. Yeah, I think you can really compare with the railway lines kind of a thing. Multiple railway lines and then interjunctions, intersections, connections, and the big station, a small station. It's so complex, especially its chemistry part, you know.
Absolutely.
Yeah. Thank you, Subhadeep. Okay.
Thank you so much. Thank you so much. Namaste to everybody. And my apologies of not being able to.
And with this, this particular session is over. Thank you very much.
Thank you very much, Session Chair Professor Ashok Puranik and speakers. Although Professor Neelam Sinha and Dr. Shubhati Bhattacharya could not be present with us physically today, we extend our heartfelt thanks to them for their valuable contribution and insightful talks. At this moment, we would like to felicitate our session chair, Professor Ashok Puranik, for his guidance and for leading this session. I would now like to invite Professor S.R. Joshi to kindly come forward and felicitate this session. Professor Ashok Puranik. Thank you, Professor Joshi and Professor Puranik. There is an announcement. There is a change of venue for the Foundation Day lecture. It will be conducted right here in Conference Hall 2 instead of auditorium. So the Foundation Day lectures will be held here itself in Conference Hall 2 instead of auditorium. So I would request everyone to kindly be seated. Thank you.
Recording stopped.
speaker for the National Academy of Science India, NASI as we call it. I'm going to, as I would call him, Chris. And he's encouraging startups and youngsters to take careers in entrepreneurship in a large manner. He's an alumnus of IIT Madras and has contributed significantly to the growth of science and technology and research, especially research in IIT Madras. He has contributed to the
Recording in progress.
To the center which is doing brain research. In addition to this, he has given a huge, large donation, and I don't want to name the money and all that, to set up the Center for Brain Research in Indian Institute of Science, Bangalore. And I mentioned these two facts because our theme symposium is on brain and cognition and so on in the mind. He is currently the chairman of the Council of the Indian Institute of Science, Bangalore. He also continues to be on several academic and research boards in the country, one of which is the Sri Chitra General Research Center in Biomedical Engineering. He is the chairman there. He is also now, a new university has come up called Nayanta University in Pune, and he's one of the co-founders of that university. And I'm also associated with it in the core group of making that happen. The theme of that is, and again I'm saying the diversity of Chris's applications, involvements is in public policy and social sciences to begin with, and it will gradually go towards the sciences also. He values research greatly. He's passionate about promoting and enhancing the level of education and research in India. He has been one of the major architects for the very prestigious prize which we know as the Infosys Prize. He may mention that in his talk. We need people like that to do the advocacy on behalf of the educational institutions and the academies with the government so that we can take greater steps for enhancing the research culture in India, especially in the youngsters. We, NASI, stand for popularization of science, and I couldn't think of anybody better to address us and to give us an idea of what is the role of academia in creating a Viksit Bharat. This was his main theme, and we are looking forward to hearing from you, Mr. Chris Gopalakrishnan. And thank you so much for taking the time to come. I know he's visiting Kaziranga, and he's driven 4 hours to give this talk and going back 4 hours. So thank you so much for your commitment to give us a talk on the Foundation Day lecture of NASI. Over to you, Chris.
Thank you, Professor Ashok Mishra. I think he should know that the most important reason I'm here is because of Ashok Mishra. Right? I can't refuse him. So when he said, you have to do this, I had to say yes only. There is only one answer possible, and if it is physically possible, I would do it. And actually, I'm not tall enough to actually see above this, but I hope I can do that. Maybe I can. No, I'll just stand here. Yeah, if you can give me the corner mic, that's better. Anyway, while he's doing that, I just want to say that I was there for the previous session also, and as Professor Mishra mentioned, I'm supporting the Center for Brain Research at IISc. I'm also supporting the Sudha Gopalakrishnan Brain Center at IIT Madras. And many of the things that I talked today in this presentation comes from my visibility into the work that's being done at these places. It clearly reinforces my view that truly world-leading, it's not world-class, world-leading research can be done in India. Truly translational research can be done in India. Sri Chitra was mentioned, Sanjay Bihari is here, the heart valve, the stent, etc., created at Sri Chitra long back, right? And then they have a series of translational work that they have done. In fact, the exhibition they did in Delhi demonstrated probably more than 25 products that they have come out with over the years actually, and continue to actually come out with. So, and then Sritosh Goswami was mentioned. The previous speaker mentioned Sritosh Goswami, who has developed a neuromorphic computing. Basically, it's an analog computing chip that they have developed. It can disrupt NVIDIA, a $4 trillion company. Disrupt. But it will take 10 years. That's the effort that we need to do. That's the money that we need to spend, and that's the effort that all of us put together must make it happen. Now, Professor Mishra was saying, advocacy from me. You will see at the conclusion of my presentation, I'm transferring that responsibility to NASI. Okay? You are the advocates. You are the people who have to make that indeed happen. All of you have to make that happen. And that's what I want to make my presentation about. NASI 1930, I just want to compliment the organization way before independence. And you stand for scientific rigor, ethics, custodian of intellectual ambition. They're all from your website only. So, you must uphold these things and you must stand for these things. Because who else can do these things? And you have to have very, very high aspirations, world class. Okay? I would like to see the presentation of the work that's being done by the people, which is world class. That's why I said Sritosh, world class. 2 Nature papers, first of a kind. Right? That's the kind of work that I would like to see happen. It can be done. I'm 150% sure it can be done because it's being done every day. I've seen many, many examples of these things. When I work with, I've gone around many IITs. Here also I saw today, the project under NMICPS, I think it's Mechanical Engineering Department, I think. I saw that. Again, truly unique, one-of-a-kind work that's being done here. That's what is feasible to do. Of course, you need to translate that and create startups and technologies. Because Viksit Bharat, a developed India, our economic superpower, will happen only through technology superiority, scientific superiority. If we don't have technology and scientific superiority, we will not become a developed nation. We will still be a lower income country. The per capita income acceleration will only happen if you own products and technologies. Okay? So that is a role, that's a responsibility our institutions must take. Because who else can do it? There is nobody else. There is nobody else to do it. I talk to industry also, they have a role to play. They need to spend some money, they need to take some responsibility. But today, I'm talking to you, and so that's why I said homework for you. I'll come back to this. The opportunity that we have, the time that is in front of us is very unique. Very, very unique. Because a confluence of multiple things are happening, which makes this time very unique, and if we don't take advantage of this, we will miss this opportunity. We have the opportunity comes from emerging technologies, multiple emerging technologies. I have a slide later which talks about so many things. AI is one of them, but quantum, genetic engineering, understanding the brain, biomedical engineering, nanotechnology, climate change creates its own challenges and opportunities, robotics, so many emerging technologies are there, and every one of them gives us an opportunity to create new leadership. Why did I choose to invest in brain sciences? Because we are not behind anybody else. Because nobody else has figured out the brain fully. So when we are not behind anybody else, if we do our work, if you work hard, if you work smartly, if you invest a little bit of money, we can probably come ahead. So we need to identify spaces we have the right to win. And we have many places where we have the right to win. And that's what we need to identify and do. Some of them we will succeed. And we should succeed in some of them. There is also a strategic importance, strategic reason for doing this, because a strong India is required for strong companies and strong institutions. If India is weak, our institutions can't be strong. And this is my message to industry. If our industry has to be strong, the country has to be strong. Right? Then only we can protect our industry and our people. So it's our responsibility to make sure that India is strong. That means strategic autonomy, technology superiority, technology, all of those things are required to create a strong India. Operation Sindoor we saw. Wars are now being fought on technology platforms. Right? Again, where will these technology platforms come? Can we depend on some other country to give this to us? It can also be shut off. Right? So we have to develop these things ourselves. And that's the reason why we need to do these things. India's position on the global stage will only come if we can take advantage of technology superiority, and the demographic dividend is also an advantage. We have a young population. In fact, every other country, the population age is increasing, the demographic dividend is going the opposite direction. India, for the next 20-25 years, we will have a young nation. That means percentage of people less than 35 is the majority segment. After 25-30 years, the percentage of people above 60 will become the majority. We'll start declining in terms of age. So next 25 years, the opportunity. That's why Viksit Bharat is very important. Right? It's not just Prime Minister saying that for good slogan, but it's the opportunity that we have to take advantage of our youth. After that, we will become an older population. This slide just shows 2000 years of economic development. India was actually, and the yellow, we were 300, 400 years because we had technology superiority. We were actually a superior knowledge economy. That again proves my postulation at the beginning, which says we need technology superiority to get economic superiority. China was. Look at the 20th century, 19th and 20th century. US came out from nowhere and became the largest economy in the world. So 100 years they could do it. Can we make 21st century India's century? Right? What made US the leader in the 20th century? Technologies that they became dominant in. Automobile revolution. The Henry Ford car revolution that they created. Radio and broadcast. TV, electrical appliances, and the second half of the 20th century, digital computers. They dominated all of these. They created huge industries, massive industries around these technologies. These are technologies that have broad implications for the society. Every home has radio, television. It's a huge economic opportunity, huge market. They invented all of them. Well, they may not have invented, but they dominated that space. Dominated that space and created massive industries. So again, because of the disruptions that are happening, everything is going to change. Can we dominate some of these spaces? And we have to be very strategic about these things. So India at crossroads, can this be India's century? There are some, few slides, so I'm just going to go through and then we can make it interactive. There are a lot of material here. I've said that this presentation can be shared to whomsoever is interested, no problem. So we prepared a report. So I have been working in the last 10 years on two things. One, can our startups ecosystem, startup ecosystem, be one of the best in the world? And I'm happy to say that we have created the third best startup ecosystem. We have $100+ billion companies valuation, many more in the pipeline. We are seen as an economy that can attract huge amount of overseas investments. So much so that our startups are becoming non-Indian, owned by non-Indians. So we have to take care of our own startups. Anyway, we have created the startup ecosystem that's an envy of the world. We can create large companies. Lenskart, for example, is the largest manufacturer of spectacles in the world. They have created a factory which is the largest in the world. Aether has, from R&D at IIT Madras, created an electric vehicle two-wheeler industry. Right? So much so, all of us are catching up now. Hero, everybody is catching up to Aether in terms of technology, in terms of ability to create this. So it's possible. These are startups. So I'm very happy to say that these deep tech startups are coming. I saw some work here, Skyroot. They're making the cone for Skyroot and things like here in IIT Guwahati. These companies are going to become very valuable companies. So I'm very happy with what is happening in the startup. We can do much more. Second, I was focusing on research. And I will come back to this, but we produced a report for Ministry of Education on how can we look at research commercialization. Along with me, IIT Gandhinagar Director, Rajat Muna, who is the co-chair for me with this. We produced this report, submitted to Ministry of Education. Some of the points I'll cover in my presentation because that's very relevant to what I want to tell you. So the three pillars of the transition from, we have, I think, scientific prowess we have in terms of papers, etc., but we want dominance. And I believe we can do that. We have to invest in research and development. This is crucial. We have to do the ecosystem transformation, and I'll talk about this. And leadership in AI is very, very important because AI is now going to be integral to even scientific research. The productivity in scientific research can accelerate 10 times with AI or more. For example, the structure of proteins, right? Previously, each protein structure was a PhD paper. Now, no more PhDs in protein structure because Google DeepMind produced all the things, I think some million protein structures, they put it in public domain. There's no need to do any more research. Why? Because of AI. Millions of protein structures are now out there in public domain. It accelerates research in a significant way. The previous speaker talked about how you can use MRI images and all that stuff, right? Clearly, AI is going to replace some human intelligent, smart work. But so be it. We have to leverage that. It's augmenting human intelligence. I always say human plus machine. We need to take advantage of this. This is not first time. Going from place A to place B, we invented of course, we first started with horses, then carts, then said automobiles, steam engine, all that stuff. Now aeroplanes, jet engines, rockets, right? We actually leverage machine to go from place A to place B in no time. So history of humankind is all about using machines to augment human capabilities. Intelligence is one of the last frontiers. Maybe there is some other frontier after that. So we need to leverage machines. And every one of these transformations will create disruptions in jobs. Again, so be it. We have to learn new skills. We have to learn new capabilities. We have to become human plus machine better than each one separately. India ranks among the top 3. So I want to first of all place on record, our achievements. Yes, we must celebrate that. But what can we do better now? Pharmaceuticals, especially in generics, we lead in the world actually. Digital infrastructure, it's amazing that by just knowing somebody's telephone, we can transfer money to them at zero cost. Again, one of the best in the world. And government is leveraging. Space program, again, I don't need to tell you, one of the 4 nations which can land on moon, land on Mars, etc. Amazing achievements. The startup ecosystem, I already talked about. But look at this. You may not be able to see this, but basically we have one of the lowest investments in R&D, 0.67% of GDP. 0.67% of GDP. And our GDP is very small, 4 trillion, right? The nations above us, United States, China, Israel, Korea, Germany, much higher. So we need to definitely increase our investments in R&D. The corporate contribution is only 0.02%. We need to increase that to 2 to 4% of GDP. You can hear us, no? Yes. I don't know, people on the, it's okay, right? Yes. So we have to increase at industry level, and I talk about it. But more importantly, I strongly feel that it's not only a funding gap, it's also an ambition gap. We do incremental research. We don't do breakthrough research. We don't set ourselves to actually think about the future. AI, for example, why are we only thinking about LLMs? Can we replace LLMs? The previous speaker talked about it's a brute force method. We throw the power of the machine at the problem. We burn the world because of the power that we are going to consume. He demonstrated that again. Shouldn't there be a better model for that? Our brain consumes 20 watts of power, 2 pounds weight. Sanjay, right? It's much better than any computer. Right? So what can we learn? How can we come out with? That's why analog computing fascinates me. Can we actually disrupt NVIDIA? Can we create an industry around that? Because, machine learning, deep machine learning is all about matrix multiplication. Analog computers are far superior in matrix multiplication than digital computers. It's like when you look at automobile, the first automobiles were electric. Then Henry Ford found that he can create a factory of automobiles using fossil fuels. And the entire world transitioned to fossil fuels. Now we are going back to electric. Digital computers is the same. In 1945, we came out with digital computers. Now we are going back to neural networks, analog computing, etc. Probably that's a better model. It definitely consumes significantly lower power. So we have to reinvent computing. This is the area where we have to invest. We may fail, but if we succeed, we'll beat the world. So we have to identify these white spaces and do significantly more investment, significantly more research. Ambition gap is where I believe we cannot forgive ourselves if we don't have the ambition. And our youth, I can see, and this is the inspiration from the startups. We talk to startup founders, they truly believe they are the best in the world. They believe they can beat anybody in the world. And if I may, and don't misunderstand me, my job is also to push you a little bit. They're all your students. They're all your students. So something you did gave them the confidence that they can beat the world. Right? We need to give that same confidence to our young scientists, that they can be the best in the world. We excel in publications, services, prototypes. We lag in high-value patents, deep tech products, and industrial scaling. We need both. We need both. So on R&D, I believe that we need to go to 3 to 4% of GDP. It can be done. And government is doing the work that it needs to do. They have created all these missions, AI mission, etc. You can argue, is it probably being spent properly, etc. But they have realized that we have to increase the spending on R&D, and they have increased the spending. Anusandhan National Research Foundation, RDI, which I am involved with, RDI fund, 1 lakh crore, $12 billion. India does not have a private sector $1 billion VC fund. And the government has taken the bold step to create a $12 billion VC fund. $12 billion VC fund from government side. And I'm now talking to high net worth individuals, private sector people, saying, sorry, you have to step up. We don't have a $1 billion VC fund, private sector. We need multiple of those. That's how our startups will stay Indian. Otherwise, it will be owned by foreigners, which is what is happening. Series B and beyond is all owned by soft tech and companies like that. So, in our report, we clearly state that our academic and research institutions, at least at the top 100, must excel in foundational research, must excel in IP ownership and commercialization. Some institutions have started doing that. Must look at deep tech startup creation and also do mission-oriented research, which is like ISRO, right? Or the Center for Brain Research I'm supporting now. One is looking at understanding Alzheimer's, dementia, Parkinson, looking at can we be part of the cure for that. Mission mode, single purpose, and large number of people working multi-year, multidisciplinary for 10 years, 15 years, 20 years on one task. IIT Madras, they want to create human brain atlas, 3D. That will be the most comprehensive, high resolution at the cellular resolution, 1 micron resolution, brain atlas, physical structure of the brain, which will be used by tomorrow surgeons, will be used to create the future MRI machines, will be used in classrooms, will be used as a reference image. If you want to see how does a normal brain image look like, we can also find out how brain develops. The first 5,000 images they published, all open science, all open source, is 5,000 images of a brain, of a fetal brain, 12 weeks to 24 weeks, at 1 micron resolution, 3D image. Journal of Comparative Neuroscience, one of the best journals in neuroscience, oldest journal I'm told, had a special supplement when this was announced. And the editor flew to India and on print said, the effort here is 10 times better than the nearest at 1/10 the cost. This is the power of India. And they will now, the entire world has done 15 brains. They have already crossed 70. They want to do 200. This is in IIT Madras. They are not a medical institution. They are not biologists. This is an engineering institute because it's a multidisciplinary thing. And now, of course, we need AI people to actually understand these images, how brain develops, etc. All kinds of things need to be done. So I feel that our top 100 institutions like IIT Guwahati must have 4 pillars now. Primarily, we started with education, then we went into research. We are doing good research, but that's not sufficient. We need to have the third leg, which is commercialization, and we need to, and startups, and the fourth leg, some focused research organizations, Center for Excellence with mission mode. All 4 must be there in our best institutions. Otherwise, we cannot support nation building. And if these institutions don't do that, who does it? Why great ideas don't reach the market? There's something called valley of death. We do great work in TRL 1, 2. We do great work now in TRL 7, 8, 9. Who does 2 to 6? Who funds 2 to 6? Philanthropy, government must step up. So slowly that is starting to happen. The missing element. There is also an expertise gap also. Infrastructure gap. Incentives gap. And I was very happy to see the work that's being done here in creating this, they were showing me how they can fabricate in metal all kinds of products and things like that. So it's actual work actually. So technology transfer labs, new creation that we need to do. We've talked about technology transfer offices, but we need not just offices, we need labs. And what I saw today was an example of a technology transfer lab. From lab prototype, they create commercial prototype, they do some customer work, they prove it, they get some customers, create a startup, and commercialization starts to happen. So pilot manufacturing, multidisciplinary teams, I think I've said target 50 TTLs across the country. Probably each IIT has maybe 2 or 3. Some ICERs, some IAC, maybe some other universities would like to get in the game. And we need to do these things. ANS, Anusandhan National Research Foundation has to do this. And part of the RDI funding will also go into this. We also need a technology cadre. Because who is going to, so there is a, talent gap here also. Because we have good scientists, we have good startup founders, the in-between people are not there now. We need to do that. Because it may not be very glamorous from an academic sense, because a professor not evaluated based on, he worked on transferring this work to commercialization. So we need to change the appraisal mechanisms, the KPIs and things like that. We have to do this. Or separate cadre has to be created, people will have to be trained, they have to be paid based on the work that they're doing, etc. Sometimes, we have to look at people, who are not just professors. The work that CBR and the SGBC in IIT Madras that I support, what I do is I said 10 years, all the people costs, our foundation will support. No questions asked. Guaranteed. So you do your science, don't worry about anything else. Somebody has to do this. Curriculum reform is also required. Design, regulation also something we need to understand. How do you cross this? Because many of the time, from lab to market, you have to cross the regulatory hurdle also. All the approvals that are required, understanding the consumer rules, regulations, and things like that. All of these things will have to be addressed here. And we have to look at, reforming the financial system, CSR rules. We need to look at government in many cases have to act as the first buyer. So that's again very, very important. So again, all of these things are in the works. Ease of doing research has become equally important like ease of doing business. And a lot of the issues that are facing scientists are now being addressed in the ease of doing research. Things like, can you buy equipment using single tender? Can you go single order? All of that is now being addressed because the directors are empowered to do some of these things. So again, this is work that we have to do it. There is nobody else to do it. The PSA is a professor like all of you. DST secretary is a professor like all of you. It's all you only. So it's together we have to make it happen. Right? And an IAS officer is also a citizen of India. He's like you and me. So together we have to make it happen because nobody else is going to do this. We have to just convince them and make it indeed happen. And I'm telling you, things are changing. I'm seeing that change happen. We need to look at valuation models, understand the economics around it. That's why I was asking questions today about, have you looked at commercial aspects of it? Can you make money doing the work that you're doing? In the lab that I visited, how do you look at the business model and things? So you need to understand. Somebody needs to understand this in the group. Otherwise, you can't take things from lab to market because it has to be commercially viable. Ultimately, to create a sustainable business, it has to be commercially viable, right? So you have to do that. I'll go through quickly. So I said our academic and research institutions are into three categories. Teaching only. There may be many institutions which are only education only, and they will be evaluated based on metrics related to education, right? Classroom feedback and student pass out ratio and fees and all that stuff. Teacher-student ratio. There will be teaching plus research where they have started doing PhDs and things like that. Paper publications will be part of that. And then they reach a certain critical mass, they go into the third, research, teaching, research and innovation. This is part of our report to Ministry of Education, and they will be evaluated. They must have this technology translation capability. They need to have an innovation ecosystem. They should be able to create startups. They should be connected with venture community. They should be connected with industry and things like that. And so we need three categories of institutions, and every institution can decide in which category they would like to be. And they will be evaluated based on those parameters. I will quickly run through this. I'm not going to spend too much time. Basically, this is extremely important for scientific research also. It's going to accelerate scientific research. We have to also incorporate AI into our teaching, all the way from schools. It's a great teaching aid. It's a tool for students. Personalized education is possible with AI tutor. How do you give quality education to the last mile, to the last student in the country? Only through technology. Today it's possible using AI. Of course, we have to think about the ethics around it. You have to think about privacy. You have to think about, biases, we have to think about fairness, all of those things we have to think about. But this is the only way we can get quality education to everybody. You can give personalized feedback to every single student. In a class of 100, you can give personalized feedback. Why not use it? We have to use it. Otherwise, somebody else is going to use it and get competitive advantage. So we better become one of the first to use it. Of course, leadership demands responsibility. I'm just going to go through this. So, Viksit Bharat 2047, a vision for developed India. This is the homework. There are 10 items here which I believe NASI should take up. Okay, R&D at 3% of GDP, 70% by industry. We need to all talk about it. Every industrialist you meet, please ask them the question, how much are you spending? Right? And say, we have to spend so much. Where are you with respect to your peers around the world, not India? Are you below or above? Please ask this question. 25 national mission labs, this is the focus research organization, etc. Actually, I should have changed this to 50 because I changed it 50 elsewhere. Shared high-performance computing AI infrastructure, we are having this, but this has to be scaled up significantly, because this is going to be again critical for the country. National Scientific Data Repository, we gave the proposal, government is doing this now. We need to scale this up very quickly because AI requires that. Now, one very important thing, please understand this. All the AI models have taken up all the publicly available data. Everything has been taken, and they completely, take data on a every second basis, whatever is publicly available, without permission. So what is next? Private data. Why do you think OpenAI is, this is being recorded, this is being broadcast, but let me say this. OpenAI is giving OpenAI free in India. All of us use our private data and use it. And what is their default setting? That data can be used to train their models. Please turn it off. All your data is being used to train their models. The next leap in AI will come when they use private data. And that's why they are giving it free. Google, I think, doesn't even ask you permission, doesn't even give you option to turn it off. OpenAI at least gives you the option to turn it off. I think. I can't find in Google where it can be turned off. Okay? We need to understand these things actually. Transform 50 universities or 100 universities, whatever you want. Please come up with a number and transform those universities to become teaching plus research plus innovation. We have to. 50,000 PhDs in industry. Industry needs to recruit PhDs and make them valuable. Otherwise, why are we creating PhDs? They are all going abroad. Right? We need to recruit them in industry. Innovation procurement fund from the government, 500 deep tech startups. That's something that we are now trying to create. In fact, 500 deep tech startups that are going to do IPO in the country. Large ones. Open science adoption. We need to look at open science and open research and things like that, so that sharing of research, etc., is possible wherever it's possible. And national inspiration programs, Professor Misra talked about Infosys Science Foundation, etc., the NASI awards, Young Scientist Awards, etc. Again, we need to create the next generation of scientists in the country. We have to inspire them to take careers in science, etc. All of these things are required. So some of these things you are doing, some of these things you must now start doing, and I'm saying NASI has to take this 10-point agenda and make that actually part of your homework and part of your agenda for the country. NASI has a role to play as advisor, advocate, and architect. And to me, the architect is very, very important. Okay? You can do this. Together, we can do this. I am not a scientist. I'm from industry. But last 10 years, I've been working with startups. I've been working with academic institutions. But I want you to now advocate this. Some of you are doing it, but I want you to do it in a much bigger way so that our institutions can be changed. It was very, very difficult. In the NMICPS program, which IIT Guwahati is there, it has taken us, it is a program for 3,600 crores. 5-year program. After 5 years, only 1,200 crores was spent. Our absorption capacity in our education institutions, very poor. After 7-8 years, there is still, I think, about 1,200 crores left. That's not good news. Right? We have to become architects for the new India that we want. That's the question. Together we have to build Viksit Bharat. I think it's our opportunity. And when I talk to, there are no many youngsters, I say that we have done a lot of the work. India has progressed significantly in the last 30 years, come a long way. But the next 30 years is going to be even more exciting, and India must become a developed economy. The responsibility rests at the youth today, the people in the age bracket maybe 30 to 50. They have to do the work, and NASI has to give the leadership to make that indeed happen. Thank you. Thank you.
The floor is open for questions. Yes.
So, I recently attended this event with the RDI fund. If you can just elaborate on it 2-3 lines, because I know what it is, but 12 billion is good, but what is it going to do?
So RDI fund is, I told you, in private sector there's no $1 billion VC fund. And hence our startups, their growth stage are dependent on foreign capital and the ownership shifts to outside India. So I tried very hard. It's not that we don't have money. I talked to family offices, I talked to industry over the last 3-4 years. We couldn't create a $1 billion fund. Every family office has created a small fund to invest in startups. But again, we don't have the mechanism to invest in large amounts of money. When it comes to growth stage, you need 30 million, 50 million, $100 million. We don't have. So government, we asked the government to set up a $1 billion fund, and hats off to the Prime Minister, his vision. He said, why stop at $1 billion? Let's create 12 billion. And I now say, shame on you industry, shame on you high net worth individual. Please set up at least $10 billion private VC funds. And they will invest in startups in the growth stage. So again, it will help deep tech startups to be Indian and list in Indian exchanges and things like that.
Sir, NEP, National Education Policy 2020, is going to be a game changer. And this is because this document has been very, very well drafted as per the needs of this country. And
ANRF is one of the outcomes of NEP.
ANRF is one of the outcomes of that policy. This NEP has many components. One of the components is 3-language formula. And you can have your higher education also in your mother tongue, or at least your primary education to middle class education will be in your own language. That will bring many brains from across the country into the mainstream of science in the future. For NEP 2020 implementation, the government has to spend a lot of money and infrastructure has to be built. If I look at the higher educational institutions at the state level, state universities, their condition is pathetic.
Yeah.
And the faculty number in the last 50 years has not increased. If I started my university with 500 faculty members, they are 500 till now after 50 years.
So
that number should multiply tenfold. So is there any provision in the government for funding that kind of situation?
So,
right, sir.
So, see, everything you shouldn't look at government for answers. Okay. That's why I said private also has to step up. So that is why I'm working with Professor Mishra to start private universities. We need to have philanthropic funding come into universities. We need to, so IIT Jodhpur, for example, the first year BTech program is done in 2 languages. Right?
Recently started, yes.
And
I come from Rajasthan.
After the first year, it seems the students are all very confident and they're able to now start learning in English. So things like this, we should now do it actually. And we have to use AI also in order to do that. So I agree. But everything government cannot be the solution. I know that's a larger discussion, but everything government should not be doing.
NEP implementation is very urgent for this country.
Well, people are doing it in, there are many
it
parts
is
of
implemented, but partly.
It
has not been implemented in letter and spirit.
No, it will take few years. See, any transition
it will take more time.
Yes. Yeah, one more question.
You want to react on my?
No. Yeah. And then the lady at the back.
My request will be, please be brief.
Brief.
Don't start.
Thank you, sir, for your inspiring lecture.
In
Can
fact,
you,
I want to recognize Mr. Rahul Mehta, who's just come in. I think he's going to follow me. So I need to respect his time also.
yeah. Sir, you mentioned about the amalgamation between industry and education. Now, just now there is a question about universities, the central universities and state universities. Are you guys thinking about having some source of funding, going to those places who are not doing great these days, but have potentials to do really good?
Yeah. See, there are too many things. Now we have to do both. We have to be democratic in all the things that we do, reach as many universities as possible, but then what happens is lowest common denominator, the funding becomes minuscule because we don't have that kind of money. So that is why I said take 100 top universities. They must be challenged to become top 100 in the world or top 50 in the world. So we need both these strategies. I was very pleased with the Institute of Eminence concept, but somehow other it was discontinued. AIMS, many of the AIMS should be actually only doing research. There should be research medical institutions. That's why I'm supporting IIT to start at this thing. So, we need hospitals, but, AIMS being at the top, we should not have all of them being just, overwhelmed with patients.
Right, correct.
Right? So, we need both strategies. We need AIMS in every city, but then will be taken over by private sector or some other thing. The AIMS has to be maybe focused on research. We don't have enough of those. Something like that. What I'm saying is we need a concentrated effort, focused effort, and we need to have a democratic effort. Both are required. Democratic effort requires private support. Maybe the government can accelerate the focused effort and get private industry to come in.
Thank you, sir.
Yeah, Dr. Sajeev Misra, please.
Okay.
Hi, Professor Vatsala Thirumalai, Dean Research from NCBS TIFR. If I may make a suggestion, if I could. We have an opportunity here with Trump being in US and the economy in European countries going down. One of the things that's happening is that many students who finish their PhDs want to come back, and India has had a paucity of postdoctoral fellows. If private partners can create attractive postdoctoral fellowships, because government funds, they are there, but money usually doesn't come on time.
For positive time, can I answer that? Yes, I completely agree. We need to do that. And I will tell you, I'm running a program with the US Consulate. From our foundation, we are bringing 12 postdocs from the US to IISc, Manipal, IIT Madras, and Anna University. 4 universities, 12 postdocs. But I insisted, I don't want anybody from Indian origin. I want non-Indians to be the 12 postdocs. I want people to come, foreigners to come to India to do research. Indian postdocs also will attract. We are doing that actually. I have a program for that. Yes.
I'm Dr. Sajeev Misra. I was the director for AIMS at Jodhpur. So we did work out a program with the IIT, the joint Masters in Medical Technology,
Yes,
taking
I know
doctors
that.
and engineers.
Yes.
So with IITs coming up and AIMS, I think, sir, that is something which can propose to government with catching them young and working together. I think that will really give a push to
I completely
there. So that is just
no. It's because the hottest area right now is at the intersection of
Healthcare and working
healthcare
together.
and AI and engineering, and that's why I said the Brain Atlas is at IIT Madras.
And they have to work together. As
Yes,
partners.
they work with 20 hospitals around the country. They supply the brains, they supply the knowledge, they supply the history. They are going to benefit from this work. So this collaboration is completely required.
Some question. While the mic is going, one of the things which is the NEP, which is not addressed, is developing proper leadership and proper governance system in the higher education system. What's your comment on that?
No, so in the proposal, what we have done is we have said select to start with 500 people, train them to become deans, leadership training. Because we don't, we assume that you're a good teacher, you automatically become a good administrator. Not true. You don't even know the rules and regulations and the policies and project management and things like that. Then we said, choose our best deans and train them to become directors. And why do an open call for directorship? From this list, choose the directors. It simplifies the whole process and you get trained people to become directors. Leadership development, training in your next level of activity is a must. Why do you believe that teachers don't need any more training? Everybody requires training.
Yes, sir.
I think NEP does address this also. NEP does address this also. I think it's there, some section is there.
I have gone through the documents, it doesn't address.
Training of teachers is important, I think.
But selection of proper vice
Yeah.
chancellors is not there.
So in our proposal, it's there now.
Okay.
Excellent talk, sir. I think one thing which is important for Viksit Bharat is our gross enrollment ratio. We are doing very poorly on that, and unless the entire country is mobilized, we will not achieve our goals.
Yeah, so yes and no. We have to also look at other systems, skilling, we have to look at vocational training. Not everybody should become an engineer. We don't need probably that many engineers and we don't need that many economists and things like that. So we have to actually now look at our requirements and come up with the educational system to suit that actually.
We'll have one last question, I see one hand there.
See, I'm only proposing these as ideas, okay? But these need to be debated, and that's why I want NASI to debate this and come up with policy papers, which then you advocate very strongly with government. Very strongly with government. What do you mean by opportunity divide? Yeah, go ahead, I can hear you.
Yeah, so there's a big difference between the opportunity provided by the different classes within the equity issue.
See, it will take time to create an equitable society. I agree today, there are still poor people, there are still illiterate people, etc. India is in 19th century, 20th century and 21st century, unfortunately. We have a big challenge ahead of us, but slowly the middle class is increasing. 50 million to 300 million to now probably close to 500 million people. So it is happening. It will happen as we become a developed economy. The faster we become a developed economy, it will be better. And that's why I'm talking about technology superiority, better quality jobs, better training, better opportunities and things like that. See, we don't have answer for everything. See, the farmers today, if you increase the productivity of farms, from 40%, only 2% of people are required. Remaining 38%, where will you put them? They don't have the education required to work in the services sector. We are a services economy. So we have to think about all these challenges, and that's why India is, challenge is huge. US developed using fossil fuels. We are constrained to develop without using fossil fuels. Otherwise, we'll kill ourselves to death, pollute the world. So we have so many constraints, but that's where constraints must spur creativity, must create innovation. We have to create a new model for development. We have to give the world a new model for development. And our model, I strongly believe, will work for 8 billion people in the world. US model is 1 billion, the top 10%. They address only the top 10%. That's all they worry about.
So how do the industry and government can play a role together doing that?
That's what I talked about, because we are creating low-cost solutions. Vaccine. The entire world is getting vaccine from India, $3 per dose. US vaccine, $30 per dose. Yeah. We can create solutions that are affordable, that are equitable, that are better. We are developing as a society using renewable sources, energy from renewable sources. We have to use nuclear. So we have to look at energy mix. We can do it. From 1.7% of our energy requirement from nuclear, we have to go to 20%. We have the technology. Now government has opened its eyes and saying private sector participation is allowed.
Okay, there are solutions. Please discuss, debate, and create an advocacy paper on these things. Thank you. Thank you all very much.
Let us all thank Dr. Krishna Gopalakrishnan.
Thank you so much for a fantastic talk. You've given us lots of food for thought how NASI can play and a role in all this, and we will definitely do that. And, and thank you for accepting our invitation. Any other questions, please write down that email and please send it. Or send it to NASI only, we'll forward it. Okay, thank you.
So I request Professor Mishra to kindly felicitate our guest, Padma Bhushan Shri Krishna Gopalakrishnan ji, with a traditional gamocha and a small token of appreciation. Thank you, sir. Now we move forward for our next guest and speaker, Shri Rahul Mehta. To do the honors, I request Professor Vinod Kumar Singh to please accompany Shri Rahul Mehta to the dais. Please give us a round of applause. So we are privileged to have with us Padma Shri Professor Vinod Kumar Singh, President of NASI, an eminent synthetic organic chemist. He is the eminent Professor Emeritus and Institute Chair in Chemistry Department at IIT Kanpur and the founding director of IISER Bhopal. He has been honored with many prestigious awards, including Shanti Swarup Bhatnagar Prize and Swarn Jayanti Fellowship. I request Professor Singh to take the proceeding forward.
Thank you. Can I speak here? It's a great honor for all of us that Sri Rahul Mehta ji is here. And on behalf of the Academy and on behalf of the Institute Organizing Committee, we would like to welcome him. Because of Indigo fiasco, he could not be here during the inauguration session. Of course, our attendance also is less than half because of Indigo fiasco. But then we had to move on and we requested him to speak to the audience something. It's online also. Basically, people are watching over Zoom. The way, of course, I don't have to say too much about Rahul Mehta ji. He's a distinguished philanthropist and chairman of Mehta Family Foundation. He is passionate about higher education system, helping India, several Indian institutes in higher education by supporting IITs and several other institutes in areas like data science, health and other things. So the way we are going to have it, it's not going to be a long session. I would like Rahul ji to speak few things, talk about his vision and say whatever he has to say.
Then I'll have some questions to say. It's more like fireside chat, but there's no fire behind it. It's totally, it's unplanned thing. So with this, I don't want to waste time. Over to you, Rahul.
Thank you.
You can speak here. Or
Can I guess you can hear that.
Yeah.
Well, so first of all, it's a privilege and an honor to be here. You know, when talking in presence of Padma Bhushan people, awardees, you know, Chris and him, and you know, it just feels like, you know, anyway, I just feel such a privilege, you know, to be invited to speak to them. Again, thank you all. I mean, I'm sure you guys are all tired, right, by the end of the day. So and the question is, how can we make it more interactive so that you all can have a much more, rather than me giving you a speech about something, I think let's figure out how to make it more and more interactive, right, because that's the easiest way. And we have a small enough crowd that actually we can have fun to make it a much more interactive conversation. But as a, you know, if you want as a background, a little bit about my background, you know, obviously, I mean, I was born in this country. I left India when I was 17 years old. You know, my parents didn't have any education. They were very poor, but they had a big dream for me, and that was to give me education. And then when I told them that I want to go abroad, they said, sure, go ahead. You know, so I went, and they said, go figure it out, right? Because they didn't know anything. So I had you know, it's a long journey, right? I remember going to, you know, in a Bombay, a place called USIS, looking through the catalogs to understand what the American university is. How do you find, you know, how do you find what a university is? How do you apply for applications? What is TOEFL? What is SAT? I mean, you got, you know, all that knowledge is now very common, but at that particular point in time, nobody even knew what a TOEFL was, or nobody knew what an SAT was, or where to apply, how do you apply. I mean, there was just a complete lack of knowledge. Then you had to figure out how to apply for a visa, how to get, you know, long story short, it took me almost 18 months to even figure out how to apply for a visa, get admissions and everything else. You know, I went to the US and I did my undergraduate. And in my undergraduate year, I wrote a small paper, and then I went to present at the first Oracle conference, and that became the basis, right? And then everybody said, when can we have that as a product? I mean, I had no idea what does that really mean, right? And that became the basis of my companies. Then I built my first company. It's called what is called as bootstrapping, if you guys don't know what it means. I've never borrowed any money. I've never taken any venture money from anybody. I just figured out, I mean, it's not like I had money from my parents. It's not like nobody could give me any money. I didn't have any loans. You know, no bank would give me loans. I didn't have any, you know, friends, family, nothing, right? I've never taken a single dime from anybody, right? Completely built my company from ground zero. Started hiring one employee, then second employee. Went on to build the company. I had global customers. I had this thing. And then it was acquired. And that was the basis of my first exit. And that is the first time in my life I had, you know, some kind of a little bit of money in my pocket. Okay. Then I went on to build 4 different companies. And then the question was, in multiple questions in life, right? At some point in time, when all your needs are met, the question is, what do you do? Okay. Could I build a 5th one? Yeah, I could build a 5th one. Would I increase my bank balance? Yeah. But is that the goal in life? No, that was not the goal. That was not my goal in life. And so from there I built. So then the question, and actually, I always believed that the funds that God had given me I was a steward of the funds, right, that God had given me. It was not for my personal consumption. So I had created the foundation at the, in the end of the first one only. I built so then I used to come here to India occasionally, and for a long time I never came because I never did my education here. I did my education abroad. So to me this was when I came back, I knew a little bit about IIT, so that's why I went to IITs, and I said, how can I help you? And the reality was a lot of complications, but I'll make it short and then we can have a questions and answers. Professor Anand at IIT Madras was the first one. He says, we're creating this School of Bioscience, would you like to fund it? And I also believed at that particular point in time that if you look at American faculty at that particular point, 50% of American faculty for most campuses were working in the field of bio, but if you look at IITs, there were not a single faculty members. And the reality becomes, then you say, well, because the same IITs were doing only the same chemical, mechanical, computer science, but they were not doing any new fields. And so the challenge is, right, so it was very easy to make a decision to fund the first School of Bioscience with the vision that the bio would expand on IIT campuses. Then you fast forward, right, and so the fundamental vision that we have for the foundation, or what we believe in, what is called as the theory of change, is that countries that create intellectual capital, right, generate economic prosperity. So the fundamental vision is that you want to create more and more intellectual capital. And what does intellectual capital mean? Well, it means we want to create bachelor's, master's, and PhD students, more and more this thing. So if you think about education and you can create more and more intellectual capital, automatically the country's economic prosperity grows. And I think, US is a great shining example of that. Why is US prosperous? It produces some of the, it has a very high amount of intellectual capital that it creates. Okay. I think so my vision was that how do I help India create the more intellectual capital? Second goal was how do I help Indian students? If you take most IITs, they still do not, in my opinion, they still do not create enough new degree programs that are necessary for the world, where the world is going. I'll take a classic example. In 2021, I spent an entire year at Stanford. And what I learned is that today most of the number one major is still computer science at Stanford, but the number two major is sustainability. You come on an Indian campus, there are no degree programs in sustainability for undergraduates. So the question becomes is that how do we get Indian education to start embracing new technologies in a way such that we can create the intellectual curiosity which will lead to economic prosperity. And then you take, the second challenge that we have that I found through my travels and interaction with various things is we spend an enormous amount of money, in research. Indian government pays for many of the PhDs, but we still do not do enough things to create what I call as a collaborative environment. Most Indian researchers still think about doing the research in a small microcosm, having some 4 or 5 PhD students, writing some papers, and they're happy with it. Right? The reality, the world has moved on. I think the science is such that no individual person can solve today's world challenges. So I think another challenge that I see is that how do we create more and more large teams, cross-disciplinary teams, interdisciplinary teams that allows this. So that's the reason why we are funding what I call as CARE conferences, which stands for Collaborative for Academic Research Excellence. Getting academics together, getting international people here, getting students to come and participate in the conference. This is how I believe we will improve Indian research ecosystem, which will translate into economic prosperity. So this is kind of like, I'm giving you a 100,000 feet view and a quick view of what we do, right? I think if you know from a philanthropy perspective, what we have done is we have funded 6 different IITs. We have created 8 schools. The IITs that we have funded today are at IIT Madras, IIT Indore, IIT Roorkee, IIT Guwahati, and Kanpur. And in the 3 areas, we have funded 4 schools of bio or health science, 3 schools of data science and AI, and 1 school of sustainability. So that's the kind of philanthropy that we have done in this thing across all the schools. What I can proudly say is what makes me most proud is that we have created new capacity, right? of 3,000 new students. That is, I feel like, our real achievement in this country, is that we're creating 3,000 new students from a capacity perspective. And just to give you an idea, just I came back from IIT Madras from yesterday, and they had our first alumni meeting for the School of Bioscience and Bioengineering, and it was so heartwarming to see the students that because of their school, so many of their lives have changed. And I think we are changing the lives of every one of the 3,000 students that graduate from that particular, from those schools, and I think that is our contribution. So let's start the questions. Is that good?
Thanks. Yeah, great. Thank you. Before I say anything, we would also like to welcome Kriti, who is accompanying Rahulji.
Yeah.
I don't know her last name. The only name I know, Kriti. So Kriti, can you
Please.
stand up,
Yeah.
and we'd like to welcome you too. Thank you. Rahulji, you already mentioned a little bit about your philanthropic
We can
journey.
do it later on. Yeah.
Can you tell us why are you giving importance to higher education in India?
I mean, to me, the reason for, basic reason is that I believe that I think we need, another way to think about it is, let me tell you, right, if you think about cancer research in this country, I don't know how many of you know about MD Anderson Cancer Institute in Houston. I feel like there are more cancer researchers at MD Anderson in Houston alone than all of India combined.
True.
Okay. And to me, the reality is a bunch of things, right? When you think about that from a statistics perspective, that just tells you that we need to create more intellectual capital, more people who understand things. B, if you think about it another way, the West is not going to solve India-specific problems. So if you think about it from a health perspective, we don't know if the prostate cancer that Indian men get are the same type of prostate cancer that the Western, this thing. And especially if you look at the last thing, what the Trump has done, right? There's not going to be any more research done on understanding the differences in whether you are an Asian, whether you are Caucasian, whether you are African American. No funding is going to be there. So I think who is going to do all that research to understand that there is a differences? The human body is going to be different, and we need to understand that. So that's the reason why I want to fund higher education in this thing.
Yeah, I also like to mention that Rahul Mehta ji, of course, he supports to have a new building and all that, but he doesn't stop here. He pays attention to small details. IIT Kanpur has built several buildings, but the best building is, which is Rahul Mehta Foundation building about the biosciences thing. So what are the challenges of Indian academia in terms of research? What do we need to do to go from research to translation? Because you had been visiting here.
Right.
And of course, you sense it, you think over, you may not be telling them, but basically you must be having some kind of challenges when you go from one IIT to another IIT.
Or tell me about it. I mean, it's very frustrating sometimes. I think that,
Let's say spade is spade. So let's
yeah,
talk freely.
I'm not trying to. I think, again, like many things in education, or there are few people on every campuses that do exceptional work. The reality becomes is, right, a large percentage of the faculty are not doing what I call as interdisciplinary thinking about large projects. And I think one of the bigger challenges I feel like we need to solve in India is also what I would call as an ambition issues. I think how do we make Indian faculty have a bigger ambition in life? And so I think to me we need to figure out that, as leaders, I mean, if whatever you as an institution can, how can we as leaders figure out how to encourage, motivate, incentivize for people to think about large multidisciplinary projects that actually solve larger problems of society. Because that, until we do that, right, I think it becomes very, we are not going to be able to try and take and do real innovative work. We are just not doing it at a scale that India needs to do for the growth that the Indian government expects. And I think we need to figure out how to crack that whole model.
And what do we need to do to go from research to translation? Because government is pushing a lot translation.
Yeah.
And of course, I have my own views, but I would like to know your views.
I think again, my personal view is that there is no, there is not one magic bullet that's going to solve it, right? Like all problems, this is going to require a lot of different efforts. And in some ways, I think the fundamental issue is, can we actually create what I would call as, we don't focus on doing research for writing papers, but research for making society better. And how do we create the right incentives to do that? I think clearly around that there has to be an ecosystem to support them. And I think to me, as institutions, we need to create a lot of different things that will enable to, I mean, if you take, and again, and I don't want to compare to the West, but I think if you were to take, there are so many support mechanisms that are there, from mentorship to how do you, in fact, if you take Stanford as an example, in fact, virtually every Stanford professor has either one or two companies. They are, they have equity stakes in multiple companies that they have started in. And there is enough incentive, there is built-in drive. And it's not, I wouldn't say that many people think of it as drive for money, but actually it's not the drive for money. It is actually the drive to make society better. And I question, to me, the issue always is, how do we get people to think big and make them feel like they need to drive to make society better, right? And I think that is a tussle that we have to figure out how to solve.
I'd just like to add here, things are changing. Although I live in India, but I cannot talk about other universities and institutions. But I know in IIT Kanpur, one can take sabbatical if you want to open your company, and large number of faculty have opened their company, number of startups has gone up. But my personal belief is, unless somebody, of course, translation is very, very important, but one cannot ignore fundamental research.
No.
So both have to go hand in hand.
Yep.
But if everyone is doing translational research without understanding what is inside, that might create more problem than it solves.
Yeah, I think so. My personal, again, like all debates, people take two ends of the spectrum. I don't think we don't need to think about that doing one research means not doing another, or doing one, I think that to me is not a good model for thinking, right? The goal should be, the model for thinking should be, how do you fundamentally, those who want to do basic research, let them do basic research. But we should also encourage those who are doing translation, can we create the ecosystem for them that can actually, enable them? Can we create the right there's the right incentives are also missing, right? And the question becomes, how do we, and not only, and when I talk about not only just financial incentives, but even think about, if you were to want to figure out how to write a contract, what is a patent attorney? How do you fundamentally get the right people in a room together? What are the right professionals that you need? The right bankers, the right, we just don't have that kind of an ecosystem that institutions are creating for the professors who are capable of doing translational work to flourish. So like I said, it's a multidimensional problem. We are not going to solve it in one day, but that's a challenge that we have to figure out.
Now, you had been traveling a lot, and of course you know India as much as we all know. What are the new areas that our country should invest on?
Okay.
Of
Big
course,
time.
you mentioned about sustainability and everybody talks about new areas, but in your mind, what should
Sure.
they doing it? And I also like to add here, although you are interacting with IIT, but IIT is not India.
Yeah.
And biological research,
Yeah.
because I had been in administration, I had been researcher, I had been administration, and I had been a policy maker. I'm still involved a lot in policy maker. I see a lot of mediocrity at top level. Forget about the lower level. If the top is mediocre, lower cannot be better than that. So areas should not be a buzzword. Just because it's the chemical engineering news, they talk about this. So in real sense, India per se, if it has to do, what are the new areas our country should invest on?
Yeah, so I think let's think about different areas, right? So for example, let's take one area that I think, and I can, those who are interested, I can share an article from New York Times. Let's take battery technologies. The amount of investment China is putting into batteries, right? The question is, how can India compete globally for, batteries and renewable? So that's one thing. If you take robotics, another $1 billion industry, we need to figure out how to get into the robotics marketplace academically. There is not a single program for robotics. Third area, if you think about it, is space. We need to create more programs in space. Space is going to become another, at least maybe $1 trillion marketplace. We got to figure out how we are going to get into space. So if you have taken, ISRO has done a lot of good things, but I think the next future is going to be a lot of work in space that's going to be translating it into actual dollars, right? I think India needs to be, and we need to get Indian students to start understanding all those things. So again, this is the reason why I feel that we need to figure out how to get Indian students to think about more than computer science, more than, they're just not that there are 2 or 3 areas where they can succeed in. The Indian undergraduate students are so bright. Question is, how can we give them the opportunities to think about new areas?
Yeah, just to add it, I just give you one data. I don't know whether you are aware of. Last year, IIT Kanpur started 4-year program, data science, statistics and data science. And program, I know it because written by my daughter. It's an all 4-year program. And JEE admission. JEE is like Gatani Gatko Loka, एक दूसरे के पीछे चलने वाली जनता. आपने कुछ किया तो पीछे वाले बिना माइंड किए सब कुछ करना शुरू कर देते हैं. After computer science, the second branch filled up, not electrical engineering, but statistics and data science. So I agree with you, students are bright, they understand what they need it. But one challenge is scale. Scale.
Yeah, absolutely.
See, our scale is very, very small.
Yep.
For example, you come, I think you live in Texas.
Yeah.
Because I have full knowledge of the area like Texas, Austin, one school of Texas, 800-acre lands, but they have 53,000 students study there. And you mentioned about one cancer hospital. So, and look at 23 IITs, I think number of students will be equal to one institute. So scale is very, very important. Of course, there are challenges which I am aware of. But one difficulty comes here about university system.
Yeah.
23 IITs, some ISRs, even NIT condition is very poor in terms of research infrastructure. But unless we strengthen our universities, it becomes very difficult to take off. Because there are times when a biology faculty wants air conditioning in biology lab, the administrator thinks, अरे हम तो अपने जमाने में बिना AC का काम किए थे. See, people talk, for example, I use toilet only in MSc. Does it mean all students should use toilet only when they go to masters? So I think we have to change. The last thing I like to point out, I did not ask question because we are running short of time. Chris Gopal Krishnan was talking about attracting postdoc undergraduate students. See, we have to understand the problem. For example, I always say in a joke, if you want to make somebody director, vice chancellor, visit his house abruptly, and if toilet and kitchen is clean, then you make them director or vice chancellor. So unless we have a hostel, it's not luxury, it's the basic needs. With the tissue paper, toilet, dry bathroom, it's very difficult to attract undergraduate students from Western country. This is my perception.
Yeah, but I disagree. I mean, first of all, my view is that first of all, we don't need students from Western countries for
He
undergraduate.
mentioned about it, that's what
I would disagree. I personally think that for undergraduates, we need to figure out, my view is, can we take, I take things like IIT Jodhpur, or take, whatever it is, IIT Indore, take the second generation IITs. They're all at 2,000 students. Can we get them to 10,000 students, right? That to me should be the number one goal. If we can't even get there, I think we got our bigger challenges. Because if you think about this, I'll give you statistics. The top number one university in China is Tsinghua University. Tsinghua produces over 50,000 engineers. That is more engineers than all IITs combined.
Yes.
So one single university in China. I think we got to get to the point where fundamentally we have to get more production, more engineering talent to compete at a global level, period. Right? I think if we can do that, we can't achieve the growth rate that India needs for the economic prosperity.
It was nice having you, Rahul Mehtaji. It's a nice.
Yeah.
Thank you very much.
Thank you. This floor is open for some question, if there are any in the audience.
Yeah. Dr. Lata Ranjan, the local organizer.
Thank you very much. I think our earlier speaker, Krish Gopalakrishnan ji, did mention that we need to have more of interdisciplinary research, and I think you are also a strong proponent and advocating the same. I think which we all agree and second to the same. Now I think there has already been a step made forward with regard to the interdisciplinary research. I think faculty scholars are coming forward, whether it is from the discipline of biosciences or bioengineering or mechanical or electrical, whatever topic we are talking about, the frontier areas of science and technology. Now my just question is this, maybe sounding quite absurd, but then I just wanted to know, is there also a cross connection between that of the great philanthropists like you and Krish Gopalakrishnan on the areas where you are also coming together and joining hands?
So, well, first of all, let me just talk about this thing. I think, on one hand, first of all, I think we need to think about interdisciplinary not just within an institution, but across institutions. And I think that is a bigger challenge to solve. Because I just find in my journey that people in, and again I use IIT as an example because that's what I know very well. People in one IIT don't even talk to people in another IIT. That's the reason why I created the CARE conferences. In last year I had a meeting of HODs of all bios, and I asked them, I said, how many of you talk to each other? And the reality was the HODs of all bio schools, all IITs, bios, ICERs, IISc, they don't even talk to each other once a year again, right? So, I mean, this is even today, this year, in fact, I came from IIT Madras, same thing, I had an HOD meeting, same things. They don't even talk. We try to create a WhatsApp group, try to get everybody to communicate. It is a huge challenge to get everybody to work on the same basis. So I think one of the goals should be, I think as a National Academy in Science should be, how can we get and incentivize all the people to talk to each other? Okay, so that's number one. Number two, I think if you ask me, Chris and I work together? I mean, that's the second question you had. To be honest with you, the answer should be, yes, we should work together. My problem is that I don't live here. I come here twice a year. And just like he came in, we know each other. He came in, obviously said, Rahul, come over to Bangalore. We should work together. I just need to figure out, in my limited time I come here, I love meeting students, spending time with students. I mean, I need to just take some time and do some other work. So I will work with Chris, no questions about that. So.
Yeah, Professor. Okay, go ahead.
Yeah, Mehtaji, we sincerely admire your groundbreaking work. and I learned that you have contributed to all the IITs. But I want to make a sincere request at this forum that why don't you please choose at least some of the universities, like across the India, you choose 5 or 10 universities to begin with and try to help them and see what is the outcome after 5 years. Because this is a since, because I am from a university background, and today I enjoy world-class facility in my institute and I'm doing very good research, I'm happy, but I know what is happening in my background. So I sincerely request you that whenever you have time, you come to India, at least 5 to 10 universities, if you give them some support and encouragement, I think the rewards will be much more in the coming years.
Not the big names.
Yeah.
No, but you to go to the universities not so well known, but there are people who are very well known within those universities and trying to do great work.
He might
Yes.
be thinking it, but the challenges he may be facing, he may not be able to tell you. Anyway, Professor Verma.
So this will
Hi,
be
Rahul.
last question
Hi,
in the
Rahul.
interest of
So
time.
nice to see you here.
Rise again, yeah.
Yeah.
I mentioned you, by the way, yesterday in IIT Madras, mentioned you about how our first Pan IIT conference and second Pan IIT conference organized by you.
Yeah, I think I know Rahul for the last 10 years. I think when we started first, so I visited his Houston facility also. He invited 6 IT professor there. So we went there and we visited them in Anderson also. We wanted to have some collaboration also, but unfortunately it did not work out because there was no facility for money for that. Yeah. And I think one day I was talking to him, I think then he said, why don't you, I gave the idea that can we have some Pan IIT conferences. I think that was the 2 times I did that, one Pan IIT conferences, and I tried to collate all the IIT biosciences people in one platform.
Yep,
I think it was wonderful. 2 session was there.
Yep.
So thank you for giving us opportunity at that IIT Madras. Now I'm not in IIT Madras. I have joined.
He's the director in IIT Allahabad.
Yeah.
Oh, okay.
So I joined here also. So, but I think he's a very kind person. That's what
No, I have one very personal question.
Yeah.
Thank you for sharing your. No, okay.
He's a surgeon, Dr. Sanjay Bihari from SGPGI Lucknow, but he's the director of Sri Chitra Institute, Trivandrum, which is going out.
We're very honored to meet you, sir. You shared your life with us. There's one striking point which you kind of just skirted over. You didn't actually give us the details. You said you've not taken any money from anyone. And your first venture, you made it a success, and then you sold it to other people, made some money, and then you started this fund. So can you please tell us what were those personal efforts that you made which actually made it a success, and how can we all be inspired by that?
I'll give you a few things. I mean, it's obviously not a simple answer because I can take 1 hour to answer your question also. I don't know that there is again, I'm trying to think about how to answer your question short. First of all, I think at the end of the day, right, I don't think, people always think success comes from money, and I don't think money is the issue, typically. I do think that if you ask me what are my key strengths that have made me successful, I would probably put relationship as number 1. Again, I don't see people as a transaction, whether it's employees. So one of the things, if you want, I can say that I'm very proud in my life is that I've never laid off anybody. Okay. Right? I've tried to fire people, but that's a different story. But I always took my responsibility to my employees seriously. If that will give you some indication of me as an insight. Like I said, I knew that every time I hired somebody, they were dependent on me, and their families were dependent on me. So I had a deep I felt a deep sense of responsibility for everybody that worked for me. I paid people more than I took out of the company. I was, and if you want as an example, I was very clear, right, that one day I realized, as an one of the person managing my finance told me, Rahul, we are running low on money, we only have 1 month's worth of salary. I went customer to customer to get the, to sell the product. And, the goal is, but I never ever had to tell my employee, I don't have money and can I pay you after 5 days? Never in my history. I don't know if that tells you anything about me.
Yeah. Okay,
So now, let us all thank Shri Rahul Mehta ji. And now I would like to ask Professor Singh to felicitate our guest and speaker, Shri Rahul Mehta ji. The token of Gamocha.
Thank you.
And a token of appreciation.
Not this one?
It's a design. No.
Short, sorry. Can you send him to the guest house?
Yes,
It's better.
Yeah, absolutely, please.
So this concludes our Foundation Day lecture.
Yeah.
I would like to remind all of you that we have cultural night planned in the auditorium, and most of the guests are already there. So in 5 minutes we can all gather in the auditorium for the cultural night. It's ready? So we can go straight away.
And dinner also, no?
Afterwards there is a dinner in the pandal. Thank you.